All right, so welcome everyone to the 8th lecture in computer architecture course. So today we'll talk about a different topic that I call intelligent genome analysis. And you already got introduced actually to this topic in the previous lectures. But today we will explain more about the hardware architectures that used for genome analysis, as well as new algorithms. So this is a fascinating topic and it's very close to my heart. I've been working on this topic for the last 7 years, and we did a lot of work in this area, but we're still very far from getting our goal achieved. So I'm Mohammed Al-Sar, and I'm working in Safari Research Group with Professor Onur Mutlu. And for today, we discuss different things. So first, we will discuss what is genome analysis. So we keep saying we would like to accelerate genome analysis, but maybe we didn't define it before. And why we need intelligent genome analysis, and what do we mean by intelligent, or the intelligence part here. And after that, we are going to analyze the genome itself and show you step by step how we analyze these genomes and infer some useful information after analyzing our genomes. So the goal of this lecture is to show how we can accelerate genome analysis. And to do that, we need to profile the entire pipeline and see what are the bottlenecks that makes genome analysis slow such that we need to accelerate it. And we find that one of the main bottleneck there is read mapping. There are other bottlenecks, but the talk will be focusing on read mapping as a main step. And after showing you the reasons why read mapping is slow, then we will explain the algorithms that we developed and the hardware accelerators to accelerate the entire pipeline or read mapping as a specific step. And towards the end of the talk, we will explain how or where's read mapping going next. What's the future of genome analysis and read mapping in specific? All right, so I really need to mention this before we start. This lecture is about how to analyze biological data using available tools. So at the end, we are computer scientists or computer architects. So we don't really know, or we don't have the good knowledge about how we analyze genomes or how we process in the wet lab. So we don't deal actually with the genomic samples, the blood or whatever, and then we analyze it in the sequencing machine. No, actually as a computer scientist or again, computer architect, what you really care is the computer science problem where you have the text data or the raw data in text format, and then you need to solve a computer science problem, and then you don't draw conclusion actually. You just generate the output and give it to the experts such that they can tell you if you have this disease or that disease or you don't have it. Okay, so now let's start first with what is genome analysis. And before we start with that, we would like to discuss the data analysis in general. So if you'd like to analyze any data, what you need to do? Do you need to generate numbers, more data, or you need to get information and useful stuff that you can leverage to draw some conclusion? So this is definition by Richard Hamming, who invented the Hamming distance, the technique of Hamming distance. And he said that the purpose of computing or analyzing is always to gain insights, not numbers. So we are not looking here for numbers or differences between the genome, but we will look further step and try to have some insights from our genome. And this is exactly what we'd like to do with the genome. So look at this picture. What you can say about genome analysis. Now, probably most of you can have some idea or some thinking about that. Genome analysis is really a way to find the cause of all these differences. So you can see the skin color is different, the hair color, even the body shape, the eye color, and so on. So what makes us healthy? What makes us unhealthy? All of this go back to our DNA. So if we check the DNA or the genome, then we might find the reason for these, and we might improve our life, make it more healthier. So that's exactly what is genome analysis in short. So genomic analysis is the identification, measurement, or comparison of genomic features such as DNA sequence or structure variation, gene expression, or even functional element annotation at genomic scale. So from this definition, you can see that there are two main aspects. One of them is structural genomics, and the other one is functional genomics. So for structural genomics, you are really looking for to build the reference, to build your own genome. So you want to find the complete sequence of your DNA such that you know the structure of this DNA, and then it will be easier for you to compare it with other DNA because you know how it looks like, what are the main components of this DNA, and so on. But for the functional genomics, you want to know exactly what's the function of each and every piece in that DNA. So some of these pieces might encode a function, can be a phenotype, can be genotype, might be something visible to you. You can see it like skin color, for example, but it might also be a disease that you are not aware, or it might not be translated into something visible to you. It might be moved from one generation to another generation, and that's what we call functional genomics. So most of the talk here, we are focusing on structural genomics rather than the functional genomics. All right, so how we do the genome analysis, or what's the first step to do that? So here's a kit that you can buy it, and this kit you can get it online, for example. This is one example that's available in Switzerland. So yeah, the promotion ends today in 12 hours. So I'm not promoting this product, but this is one of the available products in the market. You can use it to swap a genetic sample from your body and then post the sample back to the company, and they will do the analysis for you. Of course, this example is just only to know who are your parents, or whether you are coming from Africa or your origin is Europe, and so on. But if you want more features, there is another kit which is a bit more expensive than the previous one, where they can give you some information about your health. All right, so now we show you this kit that you can get the genomic sample, send it in a very convenient way, and get back your report. But now the company, what is looking for? So in the company side, they will go to your sample, try to read the chromosomes of your body, or every cell in your body. So in all cells in your body, we have 23 pairs of chromosomes, except 2 cells, which are the egg and the sperm. So in these 23 pairs of chromosomes, 22 of them are inherited from the parent, or from the mother and the father, each pair of these, or each chromosome side from one of them from the mother, one of them from the father. And we have the 23rd chromosome, which we call it 6th chromosome. So if we have XX, then this is a female candidate. And if we have X and Y in the 23rd chromosome, then this is a male candidate. And the company will go to each chromosome and try to read the DNA sequence, the exact DNA sequence, or the exact order of every pair in the DNA. And in our DNA, it's a chemical molecule. It has 4 main components, which is adenine, thymine, cytosine, and guanine. And these are connected with some phosphate backbone to make them stable, or to connect all of them together. All right, so now after we read the DNA, what will happen? What are the things that we can do with the DNA? So one of them is find the SNPs, or the genetic variation, or the differences between our DNA and other DNAs of other individuals. It could be all of them humans, for example. So here's an example for these are small pieces, small subsequences of individual DNA. So we have 16 individuals, each one is different person, and this is just a little portion of their DNA. And you have a SNP1 that the company detect for you, and we have SNP2. And then the goal here is to detect which SNP, or which genomic variation or difference that cause high blood pressure disease, for example, for some of the people. So as a smart computer scientist or computer architect, or electrical engineering student, then you might need to correlate these SNPs to the blood pressure values. So if you look at SNP2, for example, you might see, okay, we have A here, low blood pressure, we might have G here, again, still low blood pressure. We still have G, candidates with G at this location, and the blood pressure is high. But we cannot say anything about which one is causing the high blood pressure because it seems SNP2 is real random. Or at least is not correlated to the high blood pressure. But if you look at SNP1, it's real nicely correlated with the blood pressure. Whenever you have a T at this location, then it really means that you have low blood pressure. But when you have a C, then you probably will have a high blood pressure. And SNP is a single nucleotide polymorphism, which is single variation in the DNA. So some of the disease are caused by single variation, some of them are not. Now, in the next few slides, I'm going to tell you examples about those disease coming from more than a single variation. So how we do these studies typically? These studies, we call them genome-wide association study, which is in short GWAS. In GWAS, we have two groups of people. One of them, we call it as cases, and the other one, we call it as control. So cases is the one with the target disease, but the controls are healthy only in that disease. So we don't know if they are really healthy in terms of all disease or not, but at least in the target disease, these are healthy and these are unhealthy. Not necessarily disease, could be phenotype, just skin color, eye color. These are not disease, just differences because we are human and we are different in nature. So if you pick a large group of these cases and control, then you try to analyze their genome, you get the sequence of each individual genome and then try to compare them. So again, as I mentioned, we have 23 pairs of chromosomes. You pick these chromosomes and you try to map every location with other location and see how many C's are in that location, how many T's, how many A's, how many G's, and so on. And then plot these SNPs or these variation in what we call a Manhattan plot, because you can guess why we call it a Manhattan plot. You can see all these towers here. So and then we specify a threshold for the significant p-value, or let's consider it as a frequency for now. But it's a p-value where it can specify the significance of having that A, for example, is very frequent among the cases. And that's why we may say something that this disease might be caused by that SNP, by that single variation. After we specify the threshold, then we said everything above this threshold then it might be significant, such that it causes this disease and that disease. But again, as the control are not healthy in all disease, it's very hard to get such control sample. Then these SNPs need to be investigated further such that you can isolate them and study the correlation between these SNPs. Are all of them causing that disease, or some of them, or even one of them? There are other studies following exactly the same methodology. We call them TWAS for transcriptome-wide association studies and so on. So this is a paper I recommend to study, to read it, at least if you are interested in these topics. Okay, so this is a real example about the benefits that we can get from knowing these SNPs. So if you go to this link, this website, it's public, open to everyone. So you'll see a database of genetic information of individuals who did the test with some of these kits available in the market. So once you get the report back from the company showing you the differences, all the things that you have in your genome, you could voluntarily publish this information online, especially in this website, for example. And this is one of the individuals. I randomly picked that individual from the website. And then you can see very useful information. For example, at one of the location in chromosome 19, this is the exact location inside the chromosome. And at that location, so 23% of the people having their information stored in this website or database, they have T at that location, and 49% of these people having C at that location. So that can tell you the differences among, it could be a smaller population. I'm not sure how many individuals are there in the database, but the more important information is the things below down. So I told you that the chromosome is part of it coming from the mother, part of it coming from the father. So at chromosome 19, at this location, if the part coming from the mother has T at that location and the part coming inherited from the father has T again, then 20 to 25% of these people, it's related to hepatitis C patients, and 20% of those people having TT in that chromosome, they respond to treatment. So this is a very useful information for doctors, for example, so they can really decide quickly about which treatment can give you and whether you'll get benefits from such treatment or medication or not. And when you have CC, again, chromosome 19 in that location here, which is 39 million something, then if you have CC, C inherited from the mother, C inherited from the father, then 80% of such patients, hepatitis C patients, they can respond to treatment. So if you know this in advance, then that will save a lot of time, a lot of cost for the hospitalization, for example. And again, all this information are publicly available. And based on this, many of the countries realized the importance of doing a genome analysis in the hospital. Once you are admitted to the hospital and you have something serious or illness that they don't understand or they cannot map it to a known disease, then the only way to go is genome analysis. So in the UK, starting from last year, they start to do genome analysis for any seriously ill children. And that they expect to save the cost or the inpatient cost by $10,000 to $300,000 per patient. So think about how important to do this in Switzerland, for example. All right, so you think all of these diseases come from single variation? Unfortunately, no. So for example, autism and obesity. Interestingly, they are caused by an alteration of very large portion of your DNA. So it could be up to 600,000 characters that are deleted from certain chromosome, and then you might get autism, or this autism might be transferred to your kids or different generations. The same thing with obesity. So if 600,000 characters are deleted from chromosome 16, then you might have obesity. But the interesting part is that if the same portion is duplicated, then you will have totally opposite disease, which is underweight, for example. And to know more about this disease, or what we call structural variation. So we define structural variation as an alteration in your DNA by more than 50 characters. So if you have a deletion or insertion of more than 50 characters, we call them structural variation. And this paper, I find it like a really good reference, surveying all the existing methods for structural variation, tell you what are the algorithms used there. You might look there and see if there are some potential for accelerating one of these algorithms, for example, using hardware or algorithms. Okay, so I hope I covered what is genome analysis by now. And if you have any questions, just feel free to unmute yourself and just ask right away. You don't need to wait until the end of the lecture. Okay, so assume you don't have any questions. All right, so now the time is to define intelligent genome analysis. We are not looking only to analyze our genomes, but actually we want to analyze the things in an intelligent way. Okay, when you hear something intelligent, what comes to your mind? Is it the speed? Are we looking only for fast genome analysis? So yeah, it could be one of the ways, right? So we are looking, if anyone of you watched these two movies, Gattaca in 1997 and Tomorrowland in 2015, a recent one, you will see just some crazy stuff such as smartwatch where you can touch it and can analyze your DNA in just less than a second. This is exactly what we are looking for. Something like it can be used, for example, at the entrance gates in the building where they can verify your identity from the DNA rather than from something that can be hacked. So your DNA is very unique to you. So imagine something, you don't want to wait actually 2 hours to get access to a building. If you can analyze your genome in just less than 1 second, that something will be great. But again, speed is not something that will make the analysis intelligent. Speed alone will not make the genome analysis intelligent. So what else we are looking for? Are we looking for intelligent architecture? We don't really need very big devices to do this analysis for you. It might be very small devices, right? So what kind of hardware are we looking for? Is it processing inside the memory, processing inside the storage, or specialized architecture such as FPGA, GPUs, or maybe something else that we are not aware of currently? Or maybe in your smartphone, you want to have the analysis, and this is a real genome sequencer that you can give it a genetic sample here and can process it in your phone and get the data, can process it in the sequencer and give you the genomic data or the DNA, and then you can do further steps, further algorithm, and you can infer some variation between your genome and some other people genomes and get some conclusion from that in your phone. So this will be something ideal. It will be really nice to have it in such mobile devices. Okay, so now we said, okay, we want it fast, we want it intelligent in terms of architecture, very efficient, you can save energy there, you can have portable devices that don't consume that much of energy, and at the same time is cheap. But how about privacy? So in this work, this is one of our papers, we find that there are ways, whenever your DNA is sent to a third party, a company or a cloud or anyone else rather than you, then they can really know much more information than you expect. For example, think about your kids, your parents, and so on. So they can get a lot of things about the disease they may have already or they might have in the future from your own DNA. So just getting your own DNA, they can know more about you. They can even, if you don't correlate the DNA with a name, like you just publish the DNA itself without your name, they can still get more information about you. Why is that? Because from DNA, they can know your skin color, eye color, hair color, maybe the body shape, and so on. And they can correlate this with information that are published in social media, for example. From that, they can identify you. And once they identify you, it's easy to identify who's your brother, what is his eye color. And from that, they can change these places in the DNA. And at the end, they might get the full DNA of your brother, for example, or of your son, and so on. So that will create a lot of problems in the future, such that maybe the health insurance company might block you from getting a policy with their company because they know that you might have this disease that will cost them a lot in the future if you need to go to the hospital, for example. So to get more information about this work, here's the full paper. You can read it. You can have a lot of stories about real leaks in this information. People that didn't think that publishing the DNA, yeah, of course, it will help hospital, it will help doctors, it will help genetic studies to know the disease, but again, can be used against you. So there are, don't worry about that. There are too many ways to protect yourself, to protect your identity. And there are a lot of research happening in this area. So you still can contribute and publish it to help genomic studies, for example. Okay, so now we wanted privacy preserving, we want it fast, we want to have smart architecture to do genome analysis. Now, we wanted also to do rapid surveillance of disease outbreaks. For example, the COVID-19 we have, and this is for this example from Ebola in Africa. So we want something like portable. You don't need cloud computing, because in Africa, you might not have even the internet access to do these analysis and try to control the disease spread. So you want something portable. You want to do it on your own devices, small machines without large infrastructure, such that you can do it anywhere in the world. And yeah, this is a real example of where we use genome analysis for COVID-19. This is FDA approved a way of testing the COVID-19 or the coronavirus at scale for very large number of people with reasonable low cost. Okay, we want also to have population scale microbiome profiling. So think about having a very dangerous virus in Zürich, Hauptbahnhof, for example. What will happen to the people there? So it's very critical for the authorities to keep checking this on a regular basis, especially for the crowded places, to study what are the exact bacteria, viruses, or what are the things we have in these very crowded places. And we want to do this accurately, because if your tool is saying that you have this kind of virus and which is dangerous, it might cause a lot of confusion for the people. You might block this train station or whatever, you might close it, and you investigate it further, then you don't find such a virus or a bacteria. It will be a not nice thing to have. And this is one of the studies, was done in 2015 for New York City. So you can do these studies as well as not necessarily very place or single place, but you could have the same study for different places in the same city, or what we call city scale. And they did the same thing for 1,500 samples collected from all stations in New York subway system. And they find something interesting, actually. So around 50% of the things that they find was bacteria, and the other 50% something unknown. They really don't know what are these, whether they are viruses, bacteria, or something else. And more important, what they find is they found traces of a plague or Yersinia pestis bacteria. And this bacteria is real life-threatening. And for those who don't know what is it caused already death for millions of people, especially in Europe, within a few years. So this is very dangerous to have in New York, such a crowded place. Or the subway station. And this was in the media for a bit of time. And then when they investigated in the traditional way, they didn't find any traces for this bacteria. So it was a false alarm, or what we call a false positive signal. And after that, people start calling the bioinformatics as, they call this incident as a failure of bioinformatics. So it's really not nice things to have if your tool is not accurate enough to tell you the truth. So now, now we can come to a conclusion of what intelligent genome analysis can be. So there is actually a critical need for fast, and not only fast, but also accurate genome analysis. And we also have other important terms. How and where to enable fast, accurate, cheap, privacy-preserving, and exabyte scale analysis of genomic data. And I hope every and single term here, I was clear enough to explain the importance of having such terms here, especially privacy-preserving analysis, the accuracy part, the speed as well, and being able to do this at large scale. All right, do we have any questions so far? Okay, sounds good. All right, so now how we analyze genome. So we already defined it. We show the importance for intelligent genome analysis, but how we really analyze the genome. So after having this kit at your house and then pick the sample and then send this sample to the company to analyze it, what they will do, you may guess that they will take this blood sample and give you the full sequence of your DNA. But actually, we don't have any machine that can read the entire content of a genome. So don't expect that they put the blood sample in a machine and then the machine will have as output just a very large or very long string of your DNA of these character ACGTs. We don't have such a machine. And what we have is this, subsequences of your DNA. So we have machines that are very advanced, very costly, but they can give you subsequences of your DNA. And how much of these sequences we can get in how many hours, what's the cost of these pieces. So we can get 10 to the power 12 bases of these characters in 44 hours. And that will cost you about $1,000 to get. And this is what we call genome analysis. All right, so we have very different machines. They can be small in size, they can be very large, and they can generating different output. We have different specs for these machines, such as the accuracy, the read length, the cost per read, and so on. So we really have many options to perform this what we call genome sequencing. So yeah, it seems we have a question. Okay, so what's the blocker for building a machine that can read a full DNA string? That's a very good question, actually. And why we don't have such a machine that can read the entire sequence of DNA? So there are too many reasons for that. One of them, the DNA is very small, really small. So to have a machine that can read it at that scale or at that level, it's very challenging. The second thing is this DNA is double helix structure, such as we have two strands and they are connected to each other. And this DNA is super coiled around each other, such that can fit in tiny cell. So let me tell you this information. The length of the human DNA, for example, is around 3.2 billion characters. So imagine 3.2 billion chemical molecules connected to each other in very tiny cell. So imagine the skin cell, how small it can be. So it's very challenging to get a machine that can go at that very tiny level and read the full 3.2 billion chemical molecules. The main important part is that they use chemical reaction to read these bases one by one, to read if you have A, if you have C, if you have G, if you have T, and so on. So this chemical reaction will get your DNA weaker as they progress over the sequencing process. So they cannot go further up to certain length. So maybe after 100 characters in your DNA, then after that, the DNA will be damaged or will be useless. Then they need to go to another subsequence or another segment in your DNA and start reading that. So all of these challenges together lead to having such machines that they really need to chop your DNA such that can easily access these bases. But don't worry about that. The future still it seems very bright, especially that we are now having more accurate or more longer segments. And I'm going to talk about this in the next few slides. All right. Yeah, thanks. So these segments generated by the genome sequencer lack information about their order, the location, in the reference genome. So it's very important to know whether this segment coming from chromosome 19, for example, or chromosome number 1, or chromosome 2. Why is that? Because at the end, you're looking for differences that might cause a disease. So it's very important to know where's this location exactly, where's the origin of each subsequence. But these information are missing from these pieces because these pieces are basically just a string, right? Just characters. You have nothing else except just characters. So now the challenge, one of the way I'm looking into this problem is exactly as you are solving a puzzle. So you have all these pieces, which we call them read, that they are subsequences of your genome, and you try to stick them together to really connect them together such that you have the complete sequence of your DNA. And how you do that? Basically, you need something to look into it and say, okay, it seems this piece coming from that particular location, then let's stick it there. And you keep doing this for all pieces. And that's what we call a reference genome. So you need really a reference genome that is coming from somewhere, and then you try to stick your pieces based on that reference genome. However, as I said, we have really totally different machines. They are really different from each other, and they have different specs. So we can cluster them into two categories. One of them, we call them accurate, and the other we call them long. So you can guess that the accurate part are short pieces, but the long are very long, but they are not accurate. And from that, you can get, so you can guess which one is easier, to use a puzzle with 10,000 pieces, very small pieces, or you can solve a puzzle with 16 pieces, for example. Yeah, it sounds 16 pieces very easier, right? You can solve it easily because these pieces are bigger than the pattern, and the feature inside this piece can easily get matched with the reference genome. However, because of the errors, you might get some errors even in assembling these pieces together to build your genome. So what are these in numbers? So the short pieces, they are short and in the range of 100 to 300 characters. And the accuracy is real 99.9999. So they are very accurate. But the long pieces, it can be in a range of 500 characters all the way to 2 million characters. But the accuracy is only about 75%. So this is really something annoying. If you try to stick these pieces together and then you get sequencing error or a noise added by the machine rather than a genetic differences that are inherited from parent or can be from somewhere else. You cannot decide whether these differences coming from these machines or from the genetic sample itself. However, as I said, the future seems very bright. We have currently a third type of reads. We call it hi-fi reads. And in these reads, they combine the advantages of short reads and long reads. And they are long, but not as long as the long reads. They are only in 10 to 20,000 characters. Still very good to have, right? It will make the problem way easier. And they are accurate at the same time. But the problem with these reads are very expensive to have. You can go to these links down, and you can check the real example of how much this process can cost you to get these hi-fi reads. So it can be in multiple of $1,000 rather than a few $100s for short reads, for example. So don't expect that we will use these hi-fi reads at wide scales, at least in the coming years, unless the cost or the sequencing cost is going down. Imagine that you want to do this test for all the Swiss people in Switzerland, so it will cost you a lot. Imagine every individual will pay $10,000 to get the reads, for example. All right, so what we are talking about, what's the workload we have as a DNA? So for viruses, we have a few thousand characters. It's very small workload. For bacteria, we have millions of characters. So that will make the puzzle solving part is a bit challenging. How about human? As I mentioned, 3.2 billion characters. Do we think we are having the longest DNA? We are the best on Earth? Unfortunately, no. We have the red onion having 5 times what we have in our DNA. And this Japanese flower has about 150 billion characters in the DNA of these flowers. So it can be really very heavy workload to analyze in general purpose computers, for example. Okay, so I already told you about the puzzle that we need to solve, and that we need a reference picture that we can rely on to connect these pieces together. So where we get this reference genome? So the first reference genome for a human we got around 2003, and that's what we call a Human Genome Project. This project was a collaboration between many countries, including UK, US, Japan, and so on. And in this DNA, they could read not the full content of the human genome, but at some extent, yeah, they got a large portion of it. And every time and every while in a year, we get an updated version of that reference genome. Even currently, we got, I think, 3 versions in this year, updated version of the human reference genome. And in that time, in 2003, to get the 3.2 billion characters or around that, could be less because they have gaps. They couldn't read most or some of these content in the reference genome. It cost them 13 years to dig inside the human genome and get that content of the DNA, and cost them $3 billion. So imagine that every character cost them $1 to read. So this is a real example where you can get yourself the human reference genome. So the current version, we call it 38. and you can go to this link down here. You can download it yourself. It's publicly available, no restricted access, and it's compressed file. The compressed size is around 1 gig, and when you uncompress it, you will get something around 3.2 gigabyte. And this is an example of the first few characters of the file you will get. So unfortunately, we still have ends. I told you that in our DNA we have A, C, G, T, but what is N? N is something undefined. These sequencing machines still unable to read these bases. And unfortunately, we have them as the first locations, but also we have it somewhere along the reference genome. And the first line, we call it the header, and you can see that this is a chromosome 1. So you will see many of these header in the same file. Okay, now we got the reference genome. We get the genome analysis kit at our house. We pick the sample, we send it to the company. They will give you the read. It's up to you if you want to analyze it, if you have the knowledge, or the company can analyze it for you. But now at least you have the read set and you have the reference genome. So what you need to do with that? Again, you need to solve the puzzle. How you solve the puzzle? So this is the chemical format or the DNA in the row format, and then you will send it to the company. The company will give you read or pieces of your DNA, and you will have the reference genome. You start to compare every single subsequence from this read with the reference genome and say, does this piece match to that portion? Yes, okay, stick it there. No, move it to somewhere else, and so on. So you keep doing all this process until you find a good place for every single read in that reference genome. And after that, you may have a nice, smooth sequence of your DNA, but still very challenging. It's not as easy as it sounds. Okay, how about when we analyze in Zürich Hauptbahnhof, for example? If we pick a sample from there, then you don't expect to have a single species there. It's not a single DNA because many people touch, for example, the ticket machine. There are already viruses, there are already bacteria there. So what to do? What do we need to do there? So yeah, so we got a question. So let me explain this slide, and then I will tell you the answer. All right. So once we get this sample from Zürich Hauptbahnhof, for example, and we expect that we have many species, then where we can get a reference genome that can cover all these species. So the things is that we cannot find single reference genome that has all bacteria, viruses, and so on. So as you guess, or as you might guess it, then we need to have many of these reference genomes or a database of many of these species. And then we try to map every single piece in the sample we got from the company or these reads to every single reference genome we have. So you try to match these reads, then they might match partially to this reference or reference A. They again might partially match to reference B, and so on. And then at the end, you will see a lot of shared pieces from your DNA or from the sample DNA to many of these references. Then it's very challenging to decide whether what you really have in your sample. That's why in the study that was done in 2015, they find traces of Yersinia pestis, but wasn't there. And it's really not easy process to do, to infer what we have really in the sample, because again, the problem is just string matching, but at the end you are matching something not related, even if it is similar to the bacteria, but we might not have that bacteria in the first place. All right, so where does the reference genome come from? The reference genome is, yeah, it's very challenging, and I'm having something to tell you about it. Maybe we can go next slide. Yeah, so maybe I'll tell you in the next slide. I have something for you. But that's a very good way of thinking, where we can get a reference genome that can fit all the human being, for example. All right, so what are the challenges in read mapping, for example? So there are too many challenges that I already mentioned some of them while I'm explaining, but now let's have them in a list. Okay, so first thing, we need to find many mappings of each read, because the read can be short or long, it can be accurate or inaccurate, and at the same time it can match to many locations in the reference genome. And we really don't know whether this piece coming from that chromosome or that chromosome. If it really fit the same or multiple locations, then it might be worthwhile testing all these locations, for example, because all of them are, they look similar to the piece that you have in hand. And we also need to remember that the goal of genome analysis is not to read your DNA, but the goal is to find the differences. So one step to find these differences is to read your DNA, but at the end, you need to find the differences. So while you are reading DNA and constructing this puzzle and solving it, you need to tolerate the variation you have and the sequencing error in each read. So you don't look for exact matches between every piece and the reference genome as you do in puzzle. So in puzzle, it should be exactly matching there. The same piece cannot go fit here and there. So it either fit here or there, right? Same thing, or different from the solving the puzzle here, the same piece might fit in different places, and at each place we have different or multiple differences, and we need to keep these differences. So it's okay if the piece doesn't exactly match to that location, still can fit into that location. And we need to do this process very fast. Again, as I mentioned, could be life critical. Think about a kid seriously sick in the hospital, and they try many medication, none of them benefit him. So what you should do, as I mentioned, you need to do genome analysis, and from the SNPs he has in the genome, they can get some idea about what medication might be the best for him. And for that, you need to do it in really short time. And as I mentioned, the sequencers, we got these DNA pieces. We really chop the DNA into pieces, and then we start reading the DNA molecule or these bases one by one through chemical reaction. And if there's a double helix structure, you need to separate it actually, so that you can add molecules to the A, and if it interacts, then we already know that it's A, because what we add is the complement of A. So we add a chemical molecule T or thymine to the first base. If it interacts, then we said, okay, at that location it was A. And we keep repeating this process. For that, we need to split the strand. We need to split this DNA structure into two strands and start to sequence one of them or both of them. We don't really know which one coming from which after we split them. And for that, we need to map the reads to both forward and reverse strands, because we don't know what we are sequencing. Is it the forward? This, we call it forward strand, and this one, the reverse strand. They are complementing each other. But if you match the read to the forward strand, the order of the character and the content of the characters, they are different. So the read might coming from one of these at the same location, and they are just complementing each other. So if you don't compare to two strands, then you might miss some opportunity to learn the cause of a certain disease if you miss the mapping at that location. And for that, you need to do double job. So you map these reads to the forward strand, then you map them to the reverse strand. Of course, there are a lot of optimization we can do to solve this problem, to not do a double job, for example. Now, coming back to the questions. How we get the reference genome? We are, again, as I mentioned, humans are different by nature. So you don't expect to have a single reference genome that can cover all these individuals in the world. So which one to pick? We have too many reference genomes, or even if we didn't have it back in 2003, now as the sequencing cost is going down, everyone start analyzing their genome, and they already build it back based on the reference, why to not use these, what we call assembly, as a reference genome? So that's a very good way of thinking, as I mentioned, and it's already raised as a concern in many very good venues or publications. This, for example, a very good paper that studied the pan-genome or the genomes of African people, and they find that the African people have 10% more DNA compared to the current human reference genome we have. So imagine that if you are using the single reference genome that we have until now and try to study the African people, what will happen? of course, you might not get an accurate conclusions about the disease. And that would raise another question, that is it really the time to change the reference genome? So this paper showed that instead of using linear reference genome or single string to represent all individuals, we need to represent a population scale. So every 1,000 or million people, we can represent them using a single reference genome. And even going further, not even single reference genome for population, but we want to have a consensus reference. Consensus reference means we need to overlap these reference genome or the genome of each individual and try to get majority voting between the characters. If we have that character A for all of these people, then let's represent the reference genome or the consensus reference as A in that location. And we keep doing this for all of these people that are sharing some unique feature or common features, such that we can really accurately representing them using a single consensus reference or reference genome. So I hope by this I answered your question. So that's a very good question, actually. Why are we still using a single reference genome to solve the puzzle? It sounds something like crazy, right? You're looking for a small variation, single character a difference in your genome, but you are comparing with something that could be really irrelevant. Okay, so the goal of this study, again, not to teach you all these things, but actually to focus on the actual acceleration aspect for genome analysis. All right, so we have these choppers or sequencing machine. What is the throughput of this sequencing machine? How fast are these machines? So one of the latest state-of-the-art machine, we call it Illumina NovaSeq 6000. It can analyze or can sequence, can read 48 human whole genomes at 30x coverage in about 2 days. So if you put 48 samples, or assuming that can fit in one flow cell or multiple flow cells, we can analyze 48 samples in 2 days. And what is 30x coverage? So since we chop the DNA, sometimes for more accuracy, we want to have the same read or the same DNA molecule or subsequence sequenced many times. And this is what we call 30x. It means we read the same read, or hopefully the same read, 30 times. So every time the sequencing errors is totally random. So we don't expect to have it in fixed locations. So when we sequence the same subsequence many times, then we can have an idea how we can correct these reads. Because when we overlap them and we see that majority of them having A at that location, then probably the other outliers are really noise added by the sequencer. So we can ignore these and we consider A as the base at that character. So normally we use 30x as a coverage to have a good trade-off between the speed and the accuracy. But how about the softwares that are used to solve read mapping, or what we call puzzle solving step? So unfortunately, we are bottlenecked in this algorithms because we could process only one read set for a single human genome. And that will take about 32 CPU hours to be processed on 48 core processor. So it sounds really heavy. You don't really need to wait 32 CPU hours using 48, which is something like not you can have in every clinic or every genomic center, for example. So imagine in Africa, you don't have access to cluster or cloud computing in some of the places. So that will show you the need to accelerate the genome analysis at the algorithmic or at the computer side. So the sequencing machine are really doing great for that. And when we analyze what is really causing this bottleneck at the CPU side, we find that 71% of the time, which is 32 CPU hours, it's about 23 CPU hours of this time is spent only in read mapping. So the pipeline for genome analysis has many steps, and one of the early steps is read mapping, which is puzzle solving. So after you solve this puzzle, your job is not done, because you still need to infer where are the differences in the puzzle compared to the reference picture. And these steps only can be up to 29%. Now, again, it depends on the workload. These numbers might be different from one machine to another machine, from using one software or one algorithm to a new algorithm and so on, because these numbers are from 2017. But the ratio is still there. Why is that? Because these sequencing machines are also getting more and more advancement in the sequencing throughput, in the cost, and the accuracy. And as you know, Moore's law is not scaling down as good as we have in this sequencing machine. All right, so now what makes read mapper slow? So now we know that the read mapping is one of the bottlenecks. Again, it might not be the only bottleneck, but it's one of the major bottlenecks in the genome analysis pipeline. But what makes read mapping very slow? So to know what are the causes for this slowdown, we need to learn how to map a read. And once we know how we map a read, we already know what are the algorithms used for that. And then we can easily figure. I really recommend this paper. This is one of the best reference you might have for read mapping process. It tells you the entire story in about 100 pages. So it tells you everything you need to know about read mapping, and also analyze most of the existing read mappers from back to more than 20 years. So I really recommend all of you to go and read at least the first 20 pages of it. So this is a single column page, so 20 pages might be in 8 or 10 pages in double column format. Okay, so one way to solve the puzzle is really to do a brute force algorithm where you have a read coming from the sequencer. You already have the reference genome. You start to check one location by one. Does it match? Does it match? Does it match? Does it match? Until you have a good place, then you stick it there. Then you repeat the same thing for the second read, the third read, and so on. But this is a very stupid way of doing that, and it's really very expensive to do it. Why is that? Because the algorithm used to match these read and every location or every subsequence at every location, the reference genome, is very expensive. It's quadratic time algorithm. And we keep repeating this for k number of reads and n location in the reference genome. So at the end, we will get something really expensive to do. So how we can do this in a smarter way? So one way to do that is very similar to Yellow Pages. How many of you knows the Yellow Pages? You can use the yes button if you know the Yellow Pages. So you may guess it from this picture. Okay, we got 1 yes, 2 yes. Okay, it seems I'm a bit old, because when I was a kid, I was using these books to find a phone number for a person. So yeah, of course, these days we don't have Yellow Pages, but back in like 10 years back or so, or I think 10 to 15, I have one of these in my parent house, and I really look into these pages to find my uncle number or my friend number and so on. So how you search in these books? So as in any book, if you want to search for a topic, normally these books are divided into sections, and you have the index in the first few pages. And to go to a specific section, you need to know what you are looking for. So if you are looking for a name, you go to the first name and you pick the first character from the first name, and you go and find the section that all the names in that section starts with that alphabet. And once you specify which section, you go to that section, and all the names are sorted alphabetically there. And then you go to the second character in the first name, then the third character, you keep matching until you find a good match for the first name, then go to the family name and so on. So you retrieve the pages based on the section, and then you match the full name. And this This is exactly what we do in read mapping. So mapping a read is similar to querying a name or looking for a phone number in the yellow pages. Do you have any questions so far? All right, sounds good. Okay, so now let's see how they are similar. So now I told you the first step is to go to the first few pages in the book, which we call an index. And that index will tell you where to go exactly rather than looking for the entire book. So we will do the same thing for the reference genome. But the reference genome is just a string, right? Just a text, linear text. There's nothing about which page or which portion you can jump, how you can narrow down the search space, and so on. For that, we need what we call an indexing structure. So this black box can be anything, right? Anything that can tell you quickly where to jump, where to start in the reference genome. So in our paper, the paper that I recommended for you to read, which is storytelling, we find that hashing was the most popular indexing technique for read mapping since 1988, where I was born. And there are other techniques as well used for indexing, but hashing was one of the most popular indexing, and it was first used by a tool called FASTA. So let's now use the hash table for this example as well, since it's the most popular one. And how we build the hash table out of the reference genome, we divide the reference genome into pieces. We pick every piece and we convert into a hash value, or we hash it. And then we store the hash value in the buckets, and we have the location of that piece in the reference genome recorded with the hash value of it. So the first piece is green, it's here, and it's located or starts at the location number 1 here. So we record it there with number 1. We might have it also again at the location 9, 16, 30. We keep doing the same thing for all these pieces. For example, the blue piece is the second one in the reference genome. So we store it again with different hash value, of course. It might map to the same hash value, but let's assume that we have different hash values here, and it starts at location 2, for example, and so on. Of course, you don't need to store all these pieces, because at the end, we need just an index, not necessarily to tell you every page what you have there, but could be sections. So you might skip one of these pieces such that the index won't grow exponentially, for example. So every piece or every subsequence we extracted from the reference genome, we call it a seed or k-mer. So they are the same thing, seed or k-mer. If we use k-mer, then we need to specify what is k. So k, we replace it with the length of that subsequence. If we use 10 characters, then we call it 10 mers. All right, so the index, as you may guess it, similarly in the yellow page, is built only once for each reference. So once you have that reference genome, you build that index and done. You don't keep rebuilding for every sample, because the reference genome might remain the same for all these experiments. And these seeds, as I mentioned, can be overlapping, non-overlapping, spaced, adjacent, non-adjacent. There are too many theories, too many research papers about this, and I would be happy to discuss with any of you interested on how we can extract these seeds. But this is an example of what might be the workload for building the index. So the human genome, again, is 3.2 gigabyte. If you for the latest reference genome. And to build the index, it might be similar to that size, all the way to 4x as large as the human genome itself, the raw reference genome string. But also there are other tools that needs more than this for memory. It depends on how you extract these pieces. And the execution time for the indexing step also varies. So for example, Minimap2 can take about 3 minutes, but BWA-MAM, for example, can take 1 hour to process a reference genome. But again, this might not be as much as concerning, because you do this only once. But still, the index size is concerning, because you will use it for all subsequent search using a read. Whenever you have a read, you need to go to that index and search it, so that you need to load the entire index to the DRAM such that you reduce the number of misses you have. Okay, so what is the performance of human genome indexing? Now I show you only 3 examples, but this might give you a better insights about the trend we have between different algorithms used for building the index. Could be hashing, could be WT, which is Wurst-Wheeler transformation. That is a compressed index. So as I mentioned, BWT is a compressed, so the memory is always the least among all the other methods. So you can see here, I hope you can see the cursor while I'm moving, and you can see most of the tools generate or require a memory footprint that is way less than most of the other tools. But the execution time might not be the least because they need more time to compress the index. And hashing is still doing really great, especially here. You can see the memory is as good as the BWT, although it's not compressed here, but the execution time might grow way larger than the execution time of other methods. Again, the question or the debate, which one is better to use, still it depends on too many things. Are you using hardware? Are you using a CPU to process these things? Are you looking for accuracy? Are you looking for speed, and so on? So there are always trade-offs. All right, so we build the index. We already have the few pages of the yellow pages. What we should do next? So now is the time to query the name you have in your mind. So which are the reads? So we cannot query the entire read because what we store in the index are really subsequences of the entire reference genome. So we cannot search for the entire read, but the same way we build the index, we need to find subsequences in the read. So think about the first read. Now we are going to have we already have the hash table built. Now we will pick one of these seeds extracted from the read and we query the reference genome. Do you have this k-mer or seed in the hash table stored? Okay, if it is stored, what is next? Give me all the location that this seed is located in the reference genome. So for the green one, if we query it in the hash table, then we get this list of locations. So the green is located at 1, 9, 16, and 30. We do the same thing for the other pieces extracted from the read, for example, the blue piece and then the yellow piece. And for each one of them, we will get very large list of locations. However, now it's very challenging. In the yellow pages, we got normally one section to search, but here we got too many sections to search in the reference genome, but it's still much less workload than searching the entire reference genome. So you can really jump many places in the reference genome and you just search few of them. But apparently you can see that the order of these k-mers really matters because the yellow comes before the blue and the blue comes before the green piece, for example. So it makes more sense if we can search in the same order of these pieces in the original read. For example, the green here is located at location 1 in the reference genome and blue at location 2. But here the blue comes before the green. But in the reference genome, they are the opposite. So apparently this is not something that we are looking for. So you can ignore them before really investigating further and try to match the entire read to these locations. But we can see that, for example, the yellow start at 5, then the blue at 7, then the green at 9. So it sounds the same order as what we have in the read. So this is probably a good candidate to be checked or examined with the entire read content. Now we can query the hash table with substring from read to quickly find the list of possible mapping location, and that reduced the execution time a lot compared to the brute force method that I presented, or the stupid way of doing genome analysis, for example. Now, since we specify a specific location, the reference genome as a candidate, this candidate location might be large, definitely, but we still have a candidate that are shorter than having the entire reference genome to be searched. Now the time is to really find how many differences between the entire read and each subsequence in the reference genome. And for that, I told you that we use a quadratic time algorithm, but I didn't tell you what are these algorithms. So one of the algorithms used is dynamic programming algorithm, where we really investigate character by character, what is the best combination of these characters such that we make this string similar to the other string. And one example of these algorithms could be edit distance. So we are looking for the edit distance between two strings. And what is edit distance? So it's defined as the minimum number of edits. And these edits can be insertions, deletions, and or substitutions. So I told you there are diseases caused by substituting one character to another, or even deleting large portion of DNA, or duplicating or inserting large portion of your DNA. So we really need to look for all these types because we don't know in advance what we have at that location. And don't expect to have a single combination of edit operation between two strings. For example, organization and operation. If you want to calculate the edit distance between them or the number of edit operation we can do, you can see there are two different ways of doing that. So you could match the O with the O, then we have two deleted characters from here, then match, then inserting a G, then match the A, then inserting all these characters, then we have a match with all these. You could also have an alternative way of doing that, where you replace the match you have from A with another A from here. So this is another example where you can have more ways to match or to find the edit distance. Now, since we define the edit distance to be the minimum, then it might be easy to find the edit, which way give you the minimum edit distance. But again, that is not enough because we still have different ways with the same minimum number of edits to make a string exactly matching the other string. So what we should do, we use another term, we call it alignment score. The alignment score is based on the user, you can favor an insertion over a deletion. So you can give a high score for insertion and you give a lower score to deletion. And when you calculate the alignment score, the total alignment score should always be the maximum. So whenever one of these ways give you the highest alignment score, then we will stick with one of these ways. Then we said, okay, at this location in this reference genome, you have a deleted character there or inserted character there, then the experts can do a conclusion easier. Do we have any questions so far? Okay. So in our paper, we find that Smith-Waterman remains the most popular algorithm since 1988, and the Hamming distance is the second most popular. One of you might ask, Hamming distance is just the XOR operation between the pairwise characters, right? So you just compare every corresponding character with each other. So how you can find insertion and deletion? So in some of the diseases are caused by only substitutions, so it doesn't make sense to use Smith-Waterman, for example, or dynamic programming algorithms, because you are not looking for insertion or deletion. So Hamming distance suffices in this case, for example. All right, so this is one of the tools that was built by my PhD advisor, John Alakan, and Professor Onur Mutlu back in 2009, and where they use a hash table to do read mapping, for example, and it's guaranteed to find all mapping, so all location can be reported, which we call it a very sensitive read mapper, and you need to specify how many edits you are looking for when you match each read with location in the reference genome. So this tool is available online. You can download it from GitHub and you can play with the source code. So when we profile the performance of read mapping, we have different tools, and we check the CPU time and the peak memory for each tool. We find that after 2011 or 2012, most of the tools having a very fast performance, very fast CPU time, and the memory usage again is really varies, because some of the tools would like to have large hash table such that they can query more seeds faster, because the entire hash table assumed to be fit in typical memory. And by querying more seeds, you might have more execution time or less, but at the end they care about sensitivity. So they want to have, they want to find most of these locations in the reference genome, for example. So one interesting fact is that after 2013, most of these tools show consistent execution time. Why is that? Even we try now to favor the execution time over other terms. So if you correlate that with the sequencing cost, for example, you will find that at 2013, we see that the cost dropped very fast, and after that we start to have $1,000 sequencing cost. And I think by that time in 2013, we realized the need for faster genome analysis, because the cost is dropping very fast and we are generating more throughput, we are analyzing more genomes, but our CPUs are incapable of handling this much of workload. And for that, we really need just a fast read mapper or fast genome analysis tools, such that we can cope with the output of this sequencing machine, and we can find we can correlate more variation to more diseases and so on. So it's really heavy workload, especially if you want to study plant, viruses, bacteria, human being, and so on. All right, so now I already explained how we perform read mapping. Now, what makes read mapper slow? Why read mapper is slow? So we find three main observations when we profile the read mapper. First, about 90% of the execution time of read mapper, of a typical read mapper, is spent on the dynamic programming part, because you can guess querying a hash table is all one time always, assuming that it fits in the main memory. So most of the time is spent on performing the dynamic programming, or what we call sequence alignment. And when we perform this sequence alignment, we find that 98% of the comparison performed between a read and a subsequence in the reference genome, they are dissimilar. So at the end, we just ignore them. Although we perform the computation, we build the entire table for the dynamic programming, but at the end, they are dissimilar. So we need to ignore them. They are useless. We cannot draw any conclusion for these two pair of string. And only 2% of the comparison we did are useful. Now, the third observation, yeah, we are using quadratic time dynamic programming algorithm, but why we are using it if it is really expensive? So the answer might be simple as this: enumerating all possible prefixes. So we need to find the best alternative or best combination of edit operation that can make one string similar to the other. As I mentioned, we use alignment score here, not the edit distance, for example. And to find the best combination, you need to check all prefixes. Otherwise, you might miss one prefix that provide you the best alignment score, for example. So think about this example. You want to compare the word Netherlands with the Switzerland. So you pick Netherlands and you start to compare with S. This is what you perform in the second row here, for example. So S, you compare it with N, with E, with T, with H, and so on. And then you compare not with W, because every character is useless. So if you compare W, it might fit here, 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 in different location in Netherlands. But at the end, you cannot say anything about the entire read or the entire sequence. So next step is to get the two characters together, then the third character, the fourth character, and so on until you complete the entire word. And that you can see there are a lot of repeated computation because S and W are similar to S alone, then the W combined based on the S. And to save the computation, we don't really need to recompute every time. Where's S similar, then where's W similar, then what are the common between the S and W. But in fact, you can reuse the computation you did for the S, you use it for SW, and reuse the computation you use for S and W together to compute SWI, and so on. So dynamic programming sounds the best candidate to do that, because by nature, dynamic programming is designed to store the pre-computed values. So whenever you need any pre-computed values, just go up or left side and pick that value, reuse it again in later stages. Okay, so we see that there's no way to skip dynamic programming part. We need to keep using it. So now how we can accelerate it using FPGA, for example. So in FPGA, the best way to do is to divide this table, for example, and do parallelism, because the best part of FPGAs is to provide high parallelism. Unfortunately, we cannot do that because the way we build the table, at each cell, we check 3 pre-computed values, which is, let me annotate here. So if we want to compute this, we need to go check this value, and this value. And then we pick the maximum, for example, or the minimum, depends on the algorithm we're using. And then we can compute this. And same thing, when we compute this, we need to check this cell, and that cell. That will impose an order in the computation or the way you compute this dynamic programming table. So the only way to do it is that you can go, for example, row-wise, as explained here. So you build the first row, the second row, the third row, and so on. But you cannot build them in parallel, even if your hardware is capable to do that. You could do it column-wise, because when you build the first column, then when you go to the second column, these 2 values are already pre-computed, and this one already computed, so you can easily compute the second cell. You could also go anti-diagonal, and that will not violate the order imposed on the dynamic programming table. So there is data dependencies here limiting the hardware acceleration efforts. And one fact about, for example, what we call the global alignment, where you care about end-to-end comparison from really first character all the way to the last character. So in this kind of algorithm, we really need to build the entire dynamic programming table to tell you whether these having a good similarity or bad similarity, because the very last cell will tell you how many differences we have or what is the alignment score there. So we really need to compute a large portion of this table to decide whether these 2 pair of strings are similar or not. So imagine that you are already computing all of these for dissimilar sequences. What will happen? We are wasting a lot of computation to do this read mapping process. Okay, so now the most interesting part. Yeah, the previous part was interesting, but to us as computer architect, computer scientist, we really need to see some algorithms, some hardware acceleration. What's the effort to accelerate these algorithms? So is there a lower bound complexity for this problem? Yeah, sorry, I missed this question. I think you mentioned the dynamic programming, right? Or is it the read mapping in general? The dynamic programming part. Okay. So the dynamic program, yeah, I think it will be a good idea to send the questions to public so everyone can see it. Everyone can see the question, because I think I'm the only one who's seeing these questions. Yeah, so the lower bound for the dynamic programming algorithm is that when you are looking for certain disease or certain edit distance threshold, as the tool that I mentioned that was built by Jana Lakan and Onur Mutlu in 2009 and others, So in this tool, you need to specify the edit distance threshold, how many differences you are looking for between every read and every subsequence in the reference genome. So if you are looking for 4 differences, then it might not make any sense to build the entire table. So we build what we call a banded version of the dynamic programming. Banded mean we build only bands or diagonals in the dynamic programming table. So if we are looking for edit distance threshold 4, then how many bands or how many diagonals we need to build in the dynamic programming table? So we need to build the main diagonal for sure, and we need to build 4 diagonals to the right and 4 to the left. Now, why we build 4 to the right, 4 to the left, which is in total 9 diagonals in the dynamic programming table? I'm going to tell you the reason in the next section. But in short, you can see that building the banded version of this dynamic programming table save a lot of computation where you don't need to compute all of these portions. So in that case, the minimum or the lower bound on the dynamic programming computation might be somewhere between E and M or N, where M is the read length or the sequence length here, and E is the edit distance threshold. This might be better than MN, for example, where, sorry, let me write here, MN, where M is the length of one of the sequences and N is the length of the other sequence, for example. So this is in big O notation. Is this clear? Okay, yeah, the chat window is going somewhere. Yeah, whenever we use annotation with the slides, it messes with the things. So I find hard time to go back to the chat window. Let me see if it works. Okay, cool. Thank you. Yeah, so we get another question. We can get the threshold from the disease we are looking for, right? So not necessarily, especially if we don't know what we are looking for. But we might have some guesses, like if we are looking for a disease that can cause single variation, then single variation or threshold around 3, 4 might be correct. Why not 1? Because again, there are sequencing errors. So even if we are looking for single error, there are some noise added by this sequencing machine. So we cannot limit ourselves to very restricted numbers such as 1 or 2. We still need to have some flexible or wide range such that we tolerate sequencing error and the variation. And later on, once we have good overlap between many reads mapped to the same location, we can resolve or solving these sequencing errors. Then what we have left is only the, hopefully, the genetic variations. All right, yeah, these very good questions actually from all of you. So please keep it up. So couldn't we use a more naive algorithm that has less data dependencies and then exploit parallelism? Definitely, that's true. Because as I mentioned, Smith-Waterman is most widely used, but also we have Hamming distance. Hamming distance is just building the main diagonal, where you compare pairwise to each and every character between the 2 strings. So yeah, you could use Hamming distance, for example. You could accelerate it in hardware. But you could also still build hardware accelerators for Smith-Waterman. And in the next section, I'm going to tell you or show you all these algorithms, heuristics, and hardware accelerators used to accelerate this. So you have 2 ways normally. Either you go with heuristics where you compromise the accuracy, or you go with hardware accelerators where we can have parallelism, not necessarily at the cell level of the dynamic programming table, but could be solving many comparison in parallel. So you have 2 pair of string, another 2 pair of string, and these comparison are done in parallel. Although we do have built the entire dynamic programming table, but still much faster than solving them sequentially, one problem by another. So there's still a great potential for accelerating these applications. So we already know what is read mapping in details, and hopefully, do we have any questions so far for read mapping? Okay, so let's start with the accelerating efforts for read mapping. So this is the entire genome analysis pipeline, and we have the read mapping step as the bottleneck, and we try to find ways to, or what are the existing acceleration efforts that can accelerate the read mapping steps. And in this paper, which is from our lab, Safari. So we're trying with our collaborators, we're trying to survey all the existing methods such that we can understand which method might be more effective than the others. So when other people can accelerate these steps in read mapping, can find a really good way and save time by going to this approach over that approach and so on. So we find three main directions. The first direction is to accelerate indexing. Some of you might ask, indexing is done once, why we need to accelerate it? But what we mean by accelerating indexing here is to reduce the workload for the index itself, for the index data structure. So when you have a query and you want to search for it in the hash table, then you don't want many locations to be returned such that you can check one by one all of these locations. So one of the steps for accelerating the indexing is to reduce the number of seeds, for example. The other step or the other direction to accelerate the indexing is to reduce the data movement during indexing. So again, indexing is done on the CPU side, then you save the index structure, then you load it to the main memory again, and then you query, and to return the location, these locations are sent back to the CPU over several layers of caches. And to move this data back and forth is very expensive, especially if you are looking for too many seeds in the main memory. So it makes more sense to process or to verify these seeds returned from the hash table inside the memory itself, such that you can save data movement, such that you can save the energy used to move the data from different level of units in the compute stack. And you could also leverage the parallelism in DRAM or different memories to do faster indexing and faster read mapping. You could do the same thing for the next step, which is the pre-alignment filtering, what we call pre-alignment filtering. So in pre-alignment filtering, after you find potential good location, it makes more sense to not check them using dynamic programming table or dynamic programming algorithms, because you don't really care about the alignment score at this stage, because they might be dissimilar. So if they are dissimilar, you don't really care about what are the differences, what type of differences you have. Do you have insertions? Do you have deletions? Which one you favor? You favor insertion over deletion, and so on. So all of these questions, answering them at this level is not needed. So what you need to know, are they similar or dissimilar up to some extent? So they might be similar, but dissimilarity exceeding the threshold, but still better to know whether they are dissimilar, such that you save a lot of computation. And there are different ways to do pre-alignment filtering. One of them, we call it a Q-gram filtering. I'm going to show you a real example from our lab over this step. And there is the pigeonhole principle. I'm also going to show you examples over this. The other two are trivial, such as base counting. You just count how many A's, how many C's, how many G's, and how many T's you have between two strings. Based on this accounting, you can decide whether they are similar, because if they are missing one of the A's, then you could say something about it. But still, these are not accurate filters, because it might lead to very large number of false positives, for example. But they are still a good way to accelerate read mapping. For accelerating the dynamic programming step itself, you still can, as I mentioned, you could do the accurate algorithm and then build an accelerator that can solve many problems in parallel fashion, or you could reduce the computation using some heuristics, some greedy choices, such that you compromise a little bit the accuracy, but you achieve very fast computation. So you don't want to go back to the Hamming distance, and you don't want to compute a very rigorous Smith-Waterman algorithm. So you could have something in between while it's fast, but yeah, the accuracy might be affected in terms of alignment score. So maybe instead of 150, you calculate it as 120. That might be fine if you can tell me exactly where are the edits or what type of edits you have. Again, depends on the application, what you are looking for. So this is the paper. Please, I recommend it as a reading for this lecture. So please have a look on it. It's a very short paper, and you can see a lot of new things or new efforts that are recently published in the literature about how you accelerate genome analysis. So it's really a good piece of work that summarizes the existing efforts. All right. So what I'm going to describe now, most of them are the work that is published by our group, Sapphire Research Group and ETH. So we start with the first direction, accelerating the indexing step, which we call it seed filtering technique. So in seed filtering, as I mentioned, we are not aiming to accelerate building the index itself because it's done once, but we are going to reduce the number of seeds that are stored in the hash table. And definitely we are using here heuristics. These heuristics limit the number of mapping locations for each seed. And they support basically exact matches. Why is that? Because this is the nature of a hash table. This is how we build hash table. So we build it such that it can only answer queries such as, does this seed exist or not? But if I change one character, then it's totally different seed. So it cannot tell me, yeah, we have a similar seed stored here, but it's different with this character. We cannot have this information from hash table, for example. And pre-alignment filtering, we are reducing the number of invalid mapping. So we care about dissimilar sequences. If it is similar, then we just flush it to the dynamic programming algorithm. We don't do any further computation. But once we find these dissimilar sequences, then we need to pick them and remove them from the candidate pool, for example. And the pre-alignment filtering should support exact and inexact matches, because otherwise we will have a false positive or a false negative, actually, because you are deleting a good candidate. We don't want that. But it's okay to provide false positives, for example, or falsely accepted strings, because falsely accepted string, they are dissimilar, but our algorithm or pre-alignment filter says that these are similar. So it's okay to have such scenario because the dynamic programming algorithm is very accurate, 100% accurate, is very rigorous, such that can detect these and ignore them. It's okay to have few false positives at the pre-alignment filtering stage. And the last step, more details on it. You could use heuristics such as limiting the numeric range of each cell. So these cells, think about a million sequence of length, million characters. Then you will have million by million dynamic programming table, or less than that, of course, if you use banded version of it. But at the end, when you accumulate all these values cell by cell, you might end up having 16 bits numbers or 8-bit numbers. Of course, it depends on the score you assign to each insertion, deletion, and so on. So you could do some heuristics such as limiting the range for each cell, for example, or limiting the scoring function. But you could also do something like without supporting backtracking step. The backtracking step is something where we use, or in a step, we use it to tell you what are the differences and what type of differences you have based on the dynamic programming table. So once you fill the entire table, then we do what we call it a backtracking, where you go from very last cell down and you go up, you keep track which value was contributing to the optimal alignment score. And once you find this value or which cell, then you can track what are the differences between the two sequences. Now let's start with seed filtering. So this is the paper. It was published by our group in 2013 with the collaborators. And what we are aiming, or the goal of this work, is to reduce the number of seeds or seed locations that are stored in the hash table. And we use two things. So we have observation one, what we call adjacent k-mer. So the insight in this work is adjacent k-mers extracted from the read should be adjacent in the reference genome. So I already mentioned this when I was explaining the read mapping step. And the key idea is to sort the location list based on their number of locations. So sort all these location lists that are retrieved or provided by the hash table when you query the seeds. And based on the sorted location, you can know if these seeds are adjacent to the reference genome or not. And you already know that they are adjacent in the read, then you can correlate these to pick a smaller set of seeds. Here's an example. So think about this as a read, and this is a reference genome. And the read we divided into three subsequences, the red one and the yellow and the green. And this is yellow, but I don't know, it appears as brown. So now we try to query these seeds in the hash table. We get these locations. So the hash table reply back that I have this seed exists here at this location, here, here, and there. And now the yellow, the same thing, the green, the same thing. So applying adjacent k-mers from this work, it tells me the red is coming before the yellow comes before the green. And then I have the three cells or the three seeds, k-mers from the read, then they should be adjacent. So you can see here that three of them are adjacent in the correct order. We have the red, the yellow, and the green. But if you go to the next location here, you can find the red, but the yellow is very far, and the green is missing, or it's really very far from this candidate. Here, same thing. I have the red, I have the green, but I don't have the yellow. Same thing. So based on this, you can exploit this observation to reduce all these mapping location without performing any dynamic programming, just sorting the location, and that will tell you a lot about these matches. So we consider these as invalid. Now, someone might ask, we are looking for differences, right? And when we query exact matches in the hash table, we don't allow for any mismatches because we query this exactly, and this exactly, and then we require them to be adjacent here. Then where are the differences? So in this work, adjacent k-mers, they don't have to be next to each other. Adjacent could be within a range. So the green is apart from the yellow by certain number of location or certain number of bases. So you could allow that, but at least they are adjacent or nearby to each other within certain threshold. All right, so the second observation provided by this work called cheap k-mers. So there are some k-mers, we call them cheap. And what do we mean by cheap is computationally cheap to verify. So some k-mers, when we query them, the hash table will reply back by 10,000 locations, and some of them by 2 million locations, especially if these k-mers are very short. So the reference genome contains only ACGTs. So think about a very long string consists of 4 alphabets only. So the repeated subsequences are very frequent in such a very long DNA. So when you query very short subsequences, you expect to have like 2 million, 5 million, 100 million pieces of these appear in the DNA. You can observe the same thing in plants, for example, where you have much longer DNA. So the idea here is to verify only the location that's happening less than a certain threshold. So if a seed appears like 50 times, it might provide more insights than a seed happening 100,000 times. Why is that? Because at the end, you are looking for a really good match, a place where you can fit the read at that location with few mismatches. But if you find many locations, it seems this is a noisy read. It won't provide any information for me. It might still provide some information at one of these locations, but as once it's mapped to too many locations, then we consider it as a noisy. Maybe other reads will provide more benefits, more information, especially if we use more coverage. So once you cover the sequencing genome by 30x, for example, then you have 30 copies from each read. So it's okay to ignore some of these reads or some of these k-mers. Hopefully, we will recover that with the other reads and find good location with these. So the key idea is read mapper can choose the cheapest k-mer and verify their location. Let's see an example. So this is a read, and we specify the occurrence frequency to be 5 stable. So we pick the first seed around here, and we query the hash table. The hash table reply back with a location list that has only 4 locations. We keep doing the same thing with other seeds. For example, this seed, this seed, this seed, and the hash table keep replying back with different location lists. So now you can see if our occurrence threshold is 500, then we need to ignore all those having more than 500 seeds, for example, or equal to 500. It's up to you to specify this, but at least we want to find the cheap or computationally cheap k-mers. So we find that these, the 4 location, 2 location, and another 2 locations. So in total, we have around 8 locations to be verified instead of very large locations. So these are the expensive ones. We just ignore them, totally ignore them. So we saved the original location to be verified was around 3,000 locations, and now we have only 8 locations. So it sounds something reasonable to be verified, or something really fast. So all of this without involving any complex algorithm to accelerate the read mapping. And yeah, again, to recap, so the problem with existing read mapper that they perform poorly in mapping billion of short reads to the reference genome in the presence of errors. And the observation, most of the verification calculations are unnecessary because, as I mentioned, there's 98% of the comparison we are doing it for dissimilar sequences. So we need to filter them out correctly. And in this work, we only play with the seeds, their location and the seed content, such that we can filter these or leading to dissimilar sequences. And the key idea is to reduce the cost of unnecessary verification. So we have cheap k-mer and adjacent k-mer methods. And the key results out of both methods, that FastHash, which is the tool we developed, obtains up to 19x speedup over the state-of-the-art mapper without losing valid mappings. So that tells you that we don't lose any correct mapping by using these two heuristics, I would say. The paper is online, the source code is available. You can freely download it and play with it. You can think about other heuristics where you can play with the seeds, for example, such that you can perform fast read mapping. Of course, this work is a bit old, in 2013, but was a great opportunity to accelerate these things using very cheap computation. All right, so do we have any question? Yeah, please stop me if I'm fast or if something is not clear. Okay, good. So now we will go to pre-alignment filtering. So I hope by now we know that read alignment is computationally expensive, and our goal is to reduce the need for dynamic programming algorithms. How we can reduce that? We will use what we call pre-alignment filtering. So if you still remember the yellow pages or the read mapping steps, how we search in the yellow pages, so we query the hash table, we get some candidates, and then we send these candidates to the dynamic programming algorithm. So now we have a new proposed step between these two steps, which we call pre-alignment. And as it sounds, it's a step coming before the alignment step. And we have 3 main conditions for this step to work correctly. So the first condition is this step needs to filter out most of incorrect mapping, only dissimilar mapping. It's okay if it cannot filter all of them, but at least most of them. And we need to preserve all correct mapping such that we don't have false negative. And we need to do it quickly, much faster than the sequence alignment itself. Otherwise, why we add more steps if it is slowing down the entire pipeline? So we really need to have here cheap computation, such that we reduce the workload for dynamic programming significantly. So this is the first work. We call it GateKeeper. It was published in 2017 in Bioinformatics Journal. So the key observation here, if two strings differ by E edits, then every base pair match can be aligned in at most 2 E shifts. And E here is the edit distance threshold. So whenever you have E as your edit distance threshold, then you can match any character within 2 E shifts. Now you can correlate this to the information I mentioned about the banded dynamic programming, where you have 2E plus 1 diagonals. But I will give you more details in the next few slides. Okay, so the key idea based on this observation is that we need to compute a set of vectors for every pair of strings. And we call these set of vectors shifted Hamming distance vectors. And how many of these? We need 2E plus 1 Hamming vectors to identify invalid mapping. So our goal here is to identify the dissimilar sequences rather than going for the similar ones. And we try here to use only bit parallel operations such that we can nicely map the architecture to the FPGA architecture. So this is the goal, basically, to accelerate this algorithm using FPGA. Okay, what are the key results? So GateKeeper is 90x to 130x faster than SSD and the adjacency filter. The adjacency filter was used in the fast hash, the work that I just presented there, and with only 7% false positive rate. So 7% of the things that the dynamic programming need to verify only was extra. So we have about so 7% false positive rate means that we accept 7% of the dissimilar sequences to be verified by the dynamic programming, but we reject 93% of the dissimilar sequences by our filter. That's a good deal, right? Especially if you are doing it really fast using FPGA. So the addition of GateKeeper to Mr. Fast, which is the mapper that I presented and it was published in 2009 by John Alakan, Honor Motolo, and others. So if you add GateKeeper to Mr. Fast, that will result in 10x end-to-end speed up and read mapping using FPGA, of course. Okay, so what are these set of vectors that I mentioned, the 2E plus 1 vectors? So here's an example. I hope it will clear the things to compare or to provide these sets of vectors. So I would like to compare Istanbul to Istanbul. So I would like to see the differences between them. These are two exactly matching words. Now I do Hamming distance, where I do just XOR between every character. So once I do that, I find that there are 8 matches and 0 mismatches. Now let's impose ourselves an edit operation over one of these strings. Let's have a deletion. Let's delete this character A. Now the new red word Istanbul will be changed from Istanbul to Istanbul. So you'll have this new word without the character A. Now let's compare them again using Hamming distance. If we compare them, we will see only 3 matches and 5 mismatches, although we did only single operation. We just delete single character. Now how to fix that? So think about the deleted character. What effect it has? What effect it has on this word? So by deleting one character, then you shift all the trailing characters to one side, right? So they were shifted from the right to the left side. Now to fix that, we really need to bring these pieces, the trailing characters, from left to right such that we can correctly compare them to their corresponding characters there. And this is the basic idea of shifted Hamming distance. So again, let's have the 2 words and this one with the deleted A. Now we have another version of the same word, but this time it's shifted to the right. Now when we compare them using Hamming distance, we find here 3 matches, but the other version give us 4 more matches. Now how we can correlate or connect these two pieces of information, 3 matches and 4 matches. So we do XOR between them. We got the binary results. Now, apparently you need to use AND. Why AND? Because we are looking for single mismatch, which is the A here. And when you do AND operation, these zeros will cancel any things here, and these 0 will cancel any one there, and we will get single one there. That will represent the difference we have between the two strings. And when we do AND, we got this vector. So any one in this vector represents that we have a mismatch. Do we have any question here? I hope it was a very clear example showing you the set of vectors that we are generating for every 2 words. So now in this example, we generate only a single vector next to the main vector we have. Okay, we have a question. But it's not so easy with more edits. That's true. That makes sense, actually. Why is that? Because the more you generate vectors, you'll see a lot of zeros everywhere. So once you AND them together, then you might cancel the correct ones here. And I will show you another example showing this problem. But think about, for example, if you have, instead of deleting a character, if you have an insertion, for example, what will happen? How many vectors we need to generate? What if we also have a deletion and insertion at the same time? And maybe 3 insertion, 4 deletion, and substitutions. So think about all these scenarios. What will happen? So we have another question. Did you only focus on one edit? Yeah, and this example is one edit. That's why we generate one vector. But as I mentioned, we need to generalize this problem or this algorithm, we need to always build 2e plus 1. So in this way, we are always sure that we can cover any e edits, whatever these edits are, whether they are insertion, deletion, substitution, we don't care. We still can cover all of them, but we need 2e plus 1 vectors. Now, if we have an inserted character rather than deleted character, what will happen here? Let me annotate this. What will happen here? If I insert a new character, for example, T, now all these trailing characters will be shifted to the right, by 1 step. And then when I compare them, I'll get mismatches again, as I did with the deletion. So what I need to do is to have a new version where I shift this word entirely to the left. So this version, the one here, it was shifted to the right because it was deleted character. Now, if I have insertion, then I need to have another copy that is shifted to the left to cancel the effect of the inserted character. So to generalize this for one edit, I need to have one copy shifted to the right and another copy to be shifted to the left, plus the original one here. That's why I always have 2e plus 1. Plus 1 is this guy, the original one. And the e shift, they are shifted to the right, and the other e shift, they are shifted to the left. So by this, I'm covering all kind of edits, regardless whether they are insertion or deletion. Whenever I have insertion, it will be within the vectors I built here, the e vectors. Whenever I have a deletion, it will be covered by these vectors. So don't worry about this drawing, it might be messy, but I have another example for you in the next slide. Let's jump now. So this is a real example from real data. We have it in the lab. And here are the vectors. Here I'm setting the E value to be 3. So I'm generating here how many sets or how many vectors? I'm generating 7 vectors. So 2E plus 1, remember. So E here is 3. And where is the original version, which is the XOR between 2, is this guy. So what I'm calling Hamming mask. Hamming mask is just XOR between the read and the reference. That's why I'm having here a bunch of zeros. It means these are exactly matched to these. Now I have, sorry. Now I have the other E vectors. I have here 3 vectors. I'm calling them deletion mask. And you can guess why I called them deletion mask, because they can capture any deleted character. And have another E vectors where I have the, I call them insertion mask, where I can capture any inserted character. Now how this works, how I can capture this edit operation. So let's see the green segments here highlighted by this. Now, what does this green segment mean? Whenever I have a green segment on the Hamming mask, it means I have exact matches, no edit operation was before that segment. Here, although I have a difference, which is a 1, but I still see the green segment on the Hamming mask. Why is that? Because this difference is not deletion, is not insertion. What is it? It's just substitution. So you can check here, the T and G, they are replaced by each other. That's why I get 1 in the corresponding location. So that Hamming mask also covers substitution. And this is the nature of Hamming distance, right? Still valid. The Hamming distance for one vector is valid. Now, once I delete a character, what is the deleted character here? It is C. So you can see here, we have 1 C, but here we have 2 C's. So I delete one of these C's here. So what effect it has once I delete the character? All the trailing character will be shifted by left, or 1 step to the left, as I mentioned in the previous example. Now that's why all the trailing matches now appear on the second mask, which is 1 deletion mask. So whenever you see the consecutive zeros are moving between these masks, it can tell you what operation you did or what exactly the edit operation was. So if imagine the consecutive zeros, instead of having it on the 1 deletion mask, you have it on 1 insertion mask here. Then you can really infer that was an insertion before. That's why the zeros now are appearing on the shifted version, because each shifted version is canceling the effect of 1 operation. I forgot to mention how we generate these vectors. So 1 deletion mask, where we shift the query by 1 step to the right or to the left, sorry, and then we compare to the reference. The 2 deletion mask, I shift the query by 2 steps and then compare to the reference. The 3 deletion mask, I shift the query by 3 steps, all of them same direction. That's why I call it a deletion mask. And the 1 insertion mask, same thing. So 1 insertion mask where I shift the query by 1 step, but the opposite direction, and then compare to the reference. And I do the same thing to generate all these factors. Now the goal is just to find these green segments. I hope that answers your question. So you mentioned E stands for the number of edits, whatever they are. Yes, that's true. But again, it's not necessarily limiting the maximum number of edits. So what I'm limiting here is the consecutive number of edits. For example, whenever you have a deletion, it will shift the matches, these zeros, to 1 deletion mask. Now, if I have insertion again, the zeros will appear at the Hamming mask, not at the 1 insertion mask, because the insertion cancel the effect of the deletion. That's why the zeros now will appear at the Hamming mask. So you can see, even if I set the E to 1, by alternating between deletion, insertion, then I could still have maybe 10 edits, which are deletion, insertion, deletion, insertion, deletion, insertion, and so on, and still can capture those edits. So what I'm limiting here is really the consecutive, like 3 deletion in the same location, or consecutive locations, like at location 1 and 2 and 3. I delete 3 characters. Then I cannot capture 4 consecutive deletion in this example, because the first deletion will cause the zeros to appear here The second deletion will shift the next one to here. The third deletion will cause the shifted one here. But after that, if I delete more, then there is no more vector to represent this. But if I insert a character, then the zeros will go back here. I insert another character will go back here. Insert another character will go back here. Now when I insert more character, it will go back here. Now insert more character here. But if I delete character, it will go back to the previous mask. So the deletion always cancels the insertion and vice versa. How do we get the first last position of these masks? If we shift once to the right, what is the new first position? That's true. So we said once we shift these, what are the vacancy bit there? What should be 1 or 0?
You're mute.
Yeah, it seems someone muted me. Okay, so yeah, we were asking once we shift the query to the one step to the right and then compare to the reference, what will happen to the early bases that are not compared to anything? Since you don't compare them to anything, then you should represent it as 1 or mismatch because you don't have anything to compare there, right? You cannot consider it as a match because it's not a match. It's something with nothing. So we consider it as a 1. So you can see here maybe you can see it. Yeah, so we represent it as a 1 in the same place of this location. You can represent it as a 0, doesn't matter, because at the end, when you do AND operation, these will cancel each other. For example, here you have zeros, 2 zeros, 3 zeros, then they can cancel each other. And same thing when you have here zeros, 2 0, 3 0, they will cancel each other. When you have 1s, you may have zeros somewhere, or you can cancel them from the final calculation when you do the AND operation. So these are very corner cases. Yeah, it's good that you understand that you will have something in the corner cases here and there. So now our goal is to track the diagonal consecutive matches and the neighborhood map. This is what we call neighborhood map, all these factors together. How we do that? As I mentioned, we do AND operation here such that it's nicely fed to the FPGA. But remember, as we do AND operation, we see here there are 2 ones that are caused by mismatches somewhere here, and a mismatch here, which is the deletion, and the mismatch here, which is the substitution. So how many mismatches in total or edits? We have 1, 2, 3, 4. So we have 4 edits. So now, if we do AND operation, what we will get, we will get something wrong, such as this, for example. So we have 1, 2, and 3. And this is wrong. Why is that? Because the number of edits are 4. But it's okay to have the number of edits to be less than the actual edits. We don't want to overestimate. If it is 4 and we said it's 5, then we said this is dissimilar, but the sequences are similar, right? So to have less, that means false positive. It's okay to have some false positive. But also to correct that, to not cancel any of these, we did some heuristics. I'm not going to explain it here, but you can guess it here. You can cancel any what we call random zeros. So you can see here we have a lot of random zeros that have no meaning, and they are not part of these long segments of consecutive zeros. And when we cancel them, we clear the way for these ones to appear in the final bit vector, hopefully. And then we just count the number of ones here. That will tell you the number of mismatches. So let's clear all the drawings. Okay, now what are the benefits provided by having these 2e+1 vectors? So in Needleman-Wunsch, or any algorithm used for dynamic programming, such as Smith-Waterman, for example, I mentioned the data dependencies here. At each cell, I'm computing this, this, and this. But in our neighborhood map, all these cells are independent of each other. They are really pairwise comparison. C and A, they are mismatched, just add it as 1. And A and T, mismatch 1, A and A is matched, then 0. Then all of these are independent of each other. You can calculate the entire neighborhood map in one shot using single clock cycle from FPGA, for example. So by doing this, we are solving the data dependencies totally. We don't have any dependency here. And this is the equation for the dynamic programming. This is the equation for our work. All right, so after having no data dependency at all, you can accelerate it using SIMD, GPU, FPGA, or any hardware accelerators. And by having this is the first work that design pre-alignment filtering on FPGA, where we have these reads coming from the sequencer. You query the hash table, then you get some candidates for pre-alignment filtering, then you reduce really the workload significantly for the dynamic programming. So you can see the benefit of such work, that we don't really play or change or affect the dynamic programming algorithm. We don't compromise anything here, right? Everything happening as separate step that generate only some false positive, but there is no compromising for the accuracy. Now, this is how we implement it in FPGA. You all know that there are a lookup table. This lookup table can have 4 inputs, 6 inputs, 7 inputs, depends on the FPGA board or the model you are using. So we leverage this lookup table to search for the consecutive zeros and to cancel what we call the random zeros, for example. So everything is done in parallel. You can see there are many lookup tables that all working in parallel and generate the results at the same time. So all of them need 1 clock cycle to produce their results. So it's really fast to produce. And this is why we use 5 input lookup table. We believe this is the best number to be provided, but I'm not going through the technical details, but definitely I will be more than happy to discuss with any of you offline or over email or over Piazza, for example. This is the architecture where we can solve many problems on parallel. So we have parallelism at the base level for each problem, and at the bases at the pairs level as well. So we solve many problems in parallel at the same time. This is the utilization. We are utilizing a very significant less amount of the FPGA. So you can see just about 17% of the FPGA chip. You can do other computation here, for example, for sequence alignment. So you don't need to move these data from the FPGA to the main memory over PCI Express, for example. You can do sequence alignment, pre-alignment filtering, and maybe many others inside the FPGA chip itself. And these are the results which I already presented. And yeah, definitely all our design or our works are publicly available on GitHub. You can access them, download them, even the FPGA designs. Okay, so what's the conclusion for GateKeeper? So we have FPGA-based pre-alignment filter that greatly speed up read mapping, and we saw a 10x speed up of the state-of-the-art for read mapper. There's a full pipeline. And the FPGA-based pre-alignment can be integrated with the sequencer. So I didn't tell you that the sequencer uses FPGA chip. So any sequencer either has some GPUs or FPGAs to accelerate some of the process, especially where you translate the chemical reaction into bases. They use FPGA for that purpose. And since we have a low utilization for our FPGA, we still can integrate this inside the sequencer such that you don't really need to move the data outside the sequencer. You can filter most of them inside the sequencer and just provide very low workload for the user or for the expert to be matched using dynamic programming. And doing so by integrating it into the FPGA, we can help to hide the complexity and the details of the FPGA. So if anyone, if the bioinformatician or the expert in this field, such as doctors, they don't know how to use FPGA, you can have it as a black box with the sequencer. So they can just deal with one push button and they can sequence and generate the data, for example. That will enable real-time filtering while sequencing. So the sequencer always generates batches of data, not all at once. So you don't wait 44 hours, for example, to generate all these number of reads, but actually batches, and you can process batch by batch as they are generated from the sequencer. And once you are done with the last batch sequenced from the sequencer, then you can process, and nearly real time you are done with filtering as well. That's still research ideas. Anyone, we would be more than happy to work on these together. If you're interested, feel free to contact me. And this is another work from our group where we implement this in SIMD so that you can accelerate it even for CPUs without the need for FPGA. All right, so now I explained GateKeeper, and it requires 2e+1 vectors, and you need to build this entire vector, and then you do the AND operation. Now, how about scalability? Do you really need to build a vector of length 1,000,000, and you have 2e+1 of these vectors? Can you have something scalable where you can pipeline the design for FPGA for better performance, better throughput, and so on? And this is what we did in this work, which we call it Shouji. So this is a Japanese name, means sliding door. So you know Japan is famous for the sliding door in the houses. And that might tell you some insights about why we call it Shouji. So the key observation of Shouji: correct alignment always include long identical subsequences. And I think by now you already know what this means from GateKeeper. You remember these green segments that I highlighted? These are the long identical subsequences. It means that there are large subsequences shared between the two strings. And processing the entire mapping at once is ineffective for hardware design, as I mentioned. The key idea of this work to use overlapping sliding window approach to quickly and accurately find all long segments of consecutive zeros. So by using sliding window, then you are limiting yourself or the search space to be this much, then you process this area only, and then you slide your search space by a little bit, then process this only, then that only. And it will be much nicer if these search spaces are totally independent, such that you can process each search space separately and at parallel or in parallel fashion. Now, what are the key results? So Shouji is implemented on FPGA again, and to provide up to 3 orders of magnitude faster performance than its CPU implementation. And Shouji accelerate best performing CPU read aligner, EDLib, and this EDLib was published in Bioinformatics 2017 by up to 18.8x and using 16 filtering units that work in parallel. What does that mean? It means we are processing 16 problems on parallel in the same FPGA chip. And Shouji is 2.4x to 400x more accurate than GateKeeper. Why is that? I'll tell you the reason in the next slide. But remember that in GateKeeper, we do AND operation. So whenever we face a 0, it will cancel all the ones. So it might cause a lot of underestimation to the number of mismatches. Now, I always like examples. They are the best thing to explain anything rather than just keep talking for hours. So let's have an example for Shouji. And this is the neighborhood that I explained for GateKeeper. It's exactly the same, where you compare G with G, G with G, G with T, G with G, and whenever we have a match 0, mismatch is 1. Or you can think about it the other way, as I explained in GateKeeper, where we have the main diagonal as just XOR operation, and this is the 1 deletion mask. This is 2 deletion mask, 3 deletion mask, where it's generated by shifting this by 1 step, then comparing to that. It's exactly the same. But this way, it will be better for Shouji to make it anti-diagonal shape. Okay, so what is the window size? We fixed the window size to be 4. You cannot change it to 5 or 3. It should be 4. And we slide it over these diagonals. And we keep counting how many zeros, not consecutive, not necessarily consecutive, and how many zeros within each diagonal. So we have 1 0, 1 0, 1 0, 4 zeros, 1, 2 0, 1. And we said we have a greedy choice that whenever we have large number of zeros, this means this might be part of the long identical sections. or the pieces, the segments that were highlighted in green in GateKeeper. So whenever you have very large number of zeros, then it might be part of the long or the longer segment of zeros. Now, why they don't necessarily to be consecutive? Because if you have 4, then it's still consecutive, right? If you have 3, even if they are not consecutive, then still 3 of them or 2 of them are consecutive, because we limit the search space to 4. So if the consecutive thing try to reduce your computation, now you don't have to search for only consecutive. This will provide great benefits, especially for FPGAs, and I'm going to tell you why. I think I already delayed you by too much. Keep saying that. I will explain it in a few seconds. But the reason for that is just because we can use lookup table. You can look up how many zeros here using FPGA lookup table. You don't need to really have a counter and count how many zeros. You can store this pattern, and once you get this pattern in this diagonal, then you store how many zeros you have, which is 4. In this way, it will be very fast to be processed by FPGA lookup tables. Now, once we know how many zeros, we just store them somewhere. And we shift this search window by 1 step, only 1 step. So this search window overlaps with the previous search window by 3 bits or 3 cells. Now, the problem, when we already compute how many zeros we have in each diagonal, and we want to move them to here, now we will have overlapping, right? So which one to store? Is it the bits coming from the next search window or the previous one? So here again, another greedy choice that we want always to maximize the number of zeros. So what we should do, we pick the zeros that are coming from the one that increases the number of zeros. So since the next one has 3 zeros and the previous one also has 3 zeros in this place, then it doesn't matter here. So we can pick whatever we want. So we save it as it is. But if we have something that reduces the zeros, we don't store the cells at this location. So this one here doesn't affect the number of zeros, for example, so we keep it, which is part of the solution. And we keep doing this for all of them. And you can see that searching these zeros in the diagonals is totally independent of any search space or any search window that we use for this algorithm. So all of them can be parallel, and that maximizes the scalability such that this algorithm is scalable even for very long read length or reads. So even if you have 2 million long reads, then you still can process them because what you generate is only this portion, and you keep doing this for many of them. So it's pretty much very nice to have it for FPGA. And as we did in GateKeeper, we just count the number of ones. That's this is the sliding window in Japan, and what is called as Shouji in the Japanese language. Now, why we use a window of size 4? Because we said the minimum possible length of identical subsequence is to have only single zero, which means like substitution, match, then another substitution, or substitution, match, and another deletion, for example. So the minimum is to have 3. If you have 2, then you'll have wrong calculation. But why we have it as 4? We said whenever we have a green segment, for example, let's have this green color. So you have a green segment, then maybe I can make it wider. We have a green segment here, which is consecutive zeros, and another green segment. Then when you are searching in this area, then you will have 1, 0, 1, for example, and here you'll have a 0. But you might have another places in other vectors where you have starting with 1. So we don't want to take these portions that they are not part of the solution. If we know this is part of the solution and has zeros, then we will go with 4 bits such that we can search here for 0 also. Looking for this pattern will cover all possible values, especially at the corner cases between 2 segments. So we don't have any problem looking for here for 4 zeros, for example, and here for 4 zeros. But the problem will be between 2 segments, especially if they are at different bit vectors. And we draw this theoretically, actually, we analyze it, and we find really the search window of size 4 have the less false positive and that will increase the accuracy. This is how we implement it. Again, we implement it using lookup table. That's much faster than having a counter with 4 clock cycles, for example, to search for a number of zeros. And with that, we have very high performance compared to GateKeeper. And you can see the accuracy here, since we are looking for the segments instead of ANDing them. So we don't have any AND operation here. That's why the accuracy improvement is very high compared to GateKeeper, because we just look for these segments and try to count their length. Now, if we don't have, do we have any question? Am I going fast? Okay, it seems no question. So this is the last pre-alignment filter called SneakySnake. And this, we designed it for CPU, GPUs, and FPGA. So what's the observation in SneakySnake? Correct alignment, again, is a sequence of non-overlapping long matches. Now, what's the difference between this and Shouji? So here, we try to find non-overlapping matches. And what do I mean by that? So we match first the problem from string matching to single net routing problem. What do you mean by single net routing problem, which we use it in VLSI chip? So once you design a chip, you have a modules, I have a figure actually. Once you design this chip, you have some modules inside the system on chip, and you would like to connect them, for example, to VDD or to ground or to just an input that might connect all these submodules together, or an input that passes from here all the way to this pen in the chip. So this is what we call single net routing problem, where we want to connect something at this side to another thing at that side. We may also want to connect some of the modules inside. But remember, the more you collide with these modules is the less efficiency with this signal routing. So basically, you want to try to avoid as much as possible these modules on the chip. So you'd like to always detour whenever you find a module or a component in the chip, try to avoid it and go to find another route for this signal to be passed from one side to another side. Now, how is this related to string matching? Let's see the results, and then I will give you an example for that. So SneakySnake is up to 4 orders of magnitude more accurate than Shouji and GateKeeper. It's very, very nice results to have. And SneakySnake accelerates the state-of-the-art CPU-based sequence aligner, idlib, and parallel by up to order of magnitude without requiring any hardware acceleration, no GPU, no FPGA, and by 2 order of magnitudes using a hardware acceleration. Now remember, the contribution here is just pre-alignment filter. So it's really great to have something accelerating the dynamic programming by 2 orders of magnitude by only single step. So you have only add single step to the pipeline, and that will give you this effect. Now what we do, here we have something a bit different than what we have in Shouji and GateKeeper, and how we build it. So I have here the algorithm, but maybe we don't have time to go through it. But basically, we try to compare every character here by 2e+1 characters from there. Why 2e+1? Just to cover all cases, the deletion and insertion, but we do this at the base level rather than vector level. So we don't here shift and do all this stuff. So this will build you 1 column. So every column here, it means 1 character compared to e+1 characters. So more details are provided in the paper with a lot of technical details and proof that this is the correct algorithm and so on. But let's see how it works. It might be more interesting. Now, once we build this data structure, now we convert into this, which is a maze. How we convert this into this? We just convert every cell that has a value of 1 into a black box. Black box means here an obstacle, which could be the component, could be an IPs, or anything inside the chip itself that avoids the signal from getting routed or going through the chip itself. So I hope this is clear. And the yellow mean a match here, which is 0, and it means something available that you can pass through it. So this is what we call a chip maze, because we get the insights from VLSI chip design, and it looks really like a maze, and you want to avoid these black boxes. Now, again, we have an edit distance threshold of 3. Okay, that's why we have here, again, 7 vectors or 7 rows. Now, the problem becomes, this is more interesting for computer scientists because now it's totally computer science problem. So you have an entrance, you can enter from any row or any cell at the first column here, and you exit only at the last column. So you cannot exit from here, for example. And you want to let the snake pass through this maze until the exit with only avoiding 3 black boxes, 3 obstacles. So this sounds very similar to shortest path problem, where you find the shortest path from here to there, and you impose one additional restriction that you want to reduce the number of obstacles that you are facing through this path. All right, how we solve it? So we want the snake to go in some path here. How to find the path? We said, again, greedy choice. We want the path that has least number of obstacles. So we go to the first row. We want to count how many obstacles we have. We have 1, 2, 3, then some available path. Once we have an obstacle, we don't want to go through it because that will reduce the survival here. So we can survive only by 3 obstacles. And if we keep reusing them, then you are reducing your chance to survive this maze. So we would like to keep them as much as we can. So we keep counting how many obstacles, or do we have an obstacle here? Yes. Here? Yes. Here? Yes. Do we have an obstacle? Yes, we have an obstacle, but after 4 steps. So we can move 4 steps in this maze. Do we have an obstacle? Yes, after 1 step. No steps available. I face obstacle directly. And here 1 step available. So I will pick the path that has more steps to move on forward. That will give me the shortest path with less number of obstacles. And I keep doing this. Now I already have face an obstacle, so I reduce my survival times by 1, and now I will have 2 obstacles to avoid. I keep repeating the same thing row-wise, so row by row. This row has an obstacle, so I cannot pass an obstacle. Here 4 available steps, here an obstacle, here 2 available steps, an obstacle, and another obstacle. So which one to pick? Again, I will go with the greedy choice, which is this path. I'll keep repeating this until I survive with respecting these 3 obstacles. So how many obstacles I have in this path? Let's see. I have 1 here, another 1, and a 3rd one here. Do I have more obstacles? Maybe someone of you can ask, how about the vertical obstacles? Because the snake will move here, then need to move there, then go there. So for these, we don't count them. Vertically, we don't count any obstacle. Why is that? Because we are not looking here to do optimal alignment score calculation, for example, where it really matters if we move for this path or this path or this path, because I need to calculate the alignment score and then decide based on the alignment score to which path I go. In this case, yeah, I need to count vertically. But here, I just care about how many edits. I don't care if they are deletion, insertion, which path I should go up or down. I really don't care about these. And that will save the computation by a lot. And this is what makes SneakySnake even fast on CPUs. I don't need hardware accelerator actually to implement it. But always will be great to have hardware acceleration. Now, based on the way I explained the algorithm, you can see that I don't build the entire thing. And this is the great difference between this and Shouji, GateKeeper, and so on. Why is that? Because once I face an obstacle, I don't need to compute anything here. Once I face obstacle here, I stop computing. Here, 4 steps, then I continue computing. So you can see there are a lot of saved computation here, 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 and so on. That's how SneakySnake is extremely fast on just typical CPU, even without SIMD acceleration. This is implemented in pure C. There is no SMD acceleration, there is no vectorization, and so on. And everything is done on the fly. You don't need to store any calculation here. You just need to maintain the count of how many steps you can move forward, and then play with the indices. You jump from this column to this column, for example. Once you know that there are 4 steps, then you can start the indices here. Then you know there are 4 steps, then you start the indices here, and so on. These are the resource utilization for the FPGA. Again, we implemented on GPU. These are the acceleration compared to existing state-of-the-art sequence alignment for long reads. So Shouji GateKeeper doesn't work on long reads in terms of, so GateKeeper is not scalable, but Shouji is scalable, but the accuracy will degrade by too much when we go for long reads because we are looking for only short matches. But for SneakySnake, since we count the long matches as they are without any division, then that will provide great accuracy. And since we don't build the entire data structure, that will provide great speed up. And that's how we can compete even for long sequences as long as 100,000, for example, sequences. And I'm pretty sure it can scale to longer than that. So the speed up benefits we can see here, it reduces as we increase the edit distance threshold. Why is that? Because here it's sequential. This is without any hardware acceleration. All these results without hardware acceleration. So we compute the things here sequentially, row by row. We count how many obstacles, go next row, and so on. So if we accelerate it using FPGA or GPU for long reads, then we will observe great benefits as well. So this is another results for another tool, which is state of the art. And this is the paper, and GitHub, you can find all the source codes for GPU, FPGA, and CPU. All right, so now read mapping and especially filtering and sequence alignment, they are all bottlenecked by data movement. And we can see that even if you accelerate it by FPGA, if you don't accelerate the entire pipeline, then you still need to move some data from one step to another, from the CPU or from the main memory to the FPGA. So it might not be efficient to accelerate only pre-alignment filtering on FPGA, for example, because you still need to move data that will waste energy through PCI Express and will waste time by transferring the data. Even if you overlap the computation by data transfer, you're still doing it in inefficient way, actually. So what's the solution? As many of the lecture in computer architecture course, you already been introduced to the processing in memory term, where you can do the processing inside the memory itself. And that's why we need to design mapping and filtering algorithm that fit processing in memory. And that will save the data movement between the DRAM, many levels of caches, and the CPU. And in this paper, Google workload from our group, and we observed that 62% of the total system energy is spent only on data movement, and the rest is for computation. So you can see that data movement is reducing the benefits of any acceleration efforts. So what we need to do, you can move the computation close to the data. But of course, we have limited area, energy budget, and maybe the computation that we can do in DRAM, for example, is very limited. We cannot do everything at general purpose computation, for example. So we could have also specialized cores next to the data, not necessarily inside the DRAM. And this tells you that there are two ways to do processing in memory. Either changing the DRAM minimally and provide more computation power to these DRAMs, or you can use what we call 3D stacked memories. These 3D stacked memories, we have several layers of DRAM that they are stacked on top of each other, and one layer of logic layer where you can perform computation there. So you can perform computation and you have very high bandwidth of data stored in the memory. You can move them within the same die or the same chip. So this is more details on what you can do with DRAM, and this is the 3D stacked memory. Let's skip it. We don't have time for that. This is the different layer of logic and different layer of memories and one layer of logic inside the same chip. These are pointers where you can get more information about processing in memory. And let's go with Grim filter. Grim filter is a pre-alignment filter that is developed inside this 3D stacked memory. And the key idea here is that we want to reduce the data movement, and we want to exploit the high bandwidth of logic layer and this 3D stacked memory, actually. So we can perform, you can think about the logic layer as FPGA, where you can really program it for any computation you want. And the 3D stacked memory is high memory bandwidth provider, for example. And we call the algorithmic Gram filter. Gram filter with the processing in memory is 1.8x to 3.7x faster than fast hash filter across real data set. And it provides a less number of false positive. Again, this is a pre-alignment filter, so we don't want to ignore any correct pair of strings. So it's okay to have false positive, but not false negative. And how we build this filter. So we represent the data, or we enumerate all possible values coming from a string. So we consider a substring of length 4, then whatever these values can be, we store them vertically. For example, out of 4 characters, what you can build as a seed, either all of them A's, or maybe 3 A's then T, 3 A's then G, 3 A and C, for example. Then you change 2 characters, 3 characters, and so on. So you can really enumerate all of them and store them vertically as a bet vector. So whenever you have one match or a seed match coming from 2 sequences, then you can record the corresponding cell here as a 1 based on the order that I mentioned for the enumeration. So if you have a match here, then you store this 1, no match 0, match 1, and so on. Then the data layout for this arrangement is vertically, so you can access one bit vector over the logic layer and compare it with whatever you have with coming from the references. So each bit vector will represent one string. Now, maybe I can show it in the next example. Yeah, here. So this is the string from the reference genome. We do the same thing for a single read. So we pick this chunk from the reference genome, and we try to pick every 4 character or 5 or 6, doesn't matter, that will affect the length of this vector. And if we have A in this vector, then I record 1 here. If I have AAAC, then I record 1, and so on. Now I can easily compare these vectors with another vectors that represent another read, for example, or another segment, for example, here and here. All of these will be represented as just vectors and no data or seeds. That will provide great parallelism, great benefits, especially when you can compare them using just Hamming distance, right? Because now they are pairwise comparison. Is this seed exist in both sequences that I'm comparing? Is this seed exist in both sequences, and so on? So more details on this on the paper and this explanation, how we can retrieve these sequences. Then whatever we have one as a match, that will tell you how many shared k-mers between 2 sequences. If they are more than threshold, then it means that these 2 sequences are similar. If they are not similar, discard. If they are similar, send to the sequence alignment. Now, why maybe let's skip this. Why the benefits were very minimal compared to FPGA, for example, or other works such as SneakySnake. Why this end-to-end speedup is only 1.8, for example? Because we still here and do the indexing at the CPU, then we move the data to the main memory, we move just location, but we still generate these vectors for the entire reference genome and not for certain location. And then we check the entire reference genome inside the memory, and then we send back to the CPU to do sequence alignment. So if someone can do the entire pipeline inside the 3D stacked memory, then that will be more beneficial than this proposal. But this is still a great contribution given the limited bandwidth or limited logic area where you can compute the things. So the gram filter is based on simple operation. It's highly parallel because I'm having in every vertical vault, let's say, or valve on this in this 3D stacked memory chip, and I can process all of them in parallel, and definitely is memory bound. How many I can fit on these, how many I can read from the memory is will specify how many filtering unit I can use. So this is more details about the paper, and this is more about accelerating sequence alignment. GenCache, it's a work where they accelerate sequence alignment inside the cache itself. So no processing in memory or processing in CPU or specialized accelerators. This is inside the cache, and they provide great benefits. It's 1.3x faster than going with a specialized chip, which is GenX. So GenX, you can have processing in memory and specialized chip as well. So yeah, this is in terms of CPU because they provide many levels of pre-alignment filtering. So they use SSD, for example, which is our work, as one of the pre-alignment filtering, but they use other pre-alignment filters as well at different stages. So pre-alignment filter, for example, SSD or GateKeeper or SneakySnake can provide filtering for certain type of strings or limited filtering capability for different type of string. So if you combine many of these, you can provide benefits for different filtering. You can filter a group of string here, then another group there, and third group there. Now this is the comparison, the result of this, and this is a Darwin customized chip that can provide acceleration for sequence alignment, but they use heuristics to divide the dynamic programming into sub-regions. So let me go fast over this. Yeah, this again, I already explained the dynamic programming, but here what they do is that they divide the dynamic programming into independent or with little dependencies between these. So you can see the dependencies only in this overlapping region. And now they can accelerate the computation by reducing the number of cells. Now, whether this is accurate or inaccurate or it's optimal or heuristic is another question. So they provide in the paper insights that if you increase the overlapping and you increase the size of this subproblem, you might receive some optimal results for some of the data sets. And this is the speed up you can provide by reducing the amount of workload in every dynamic programming table. Now, don't worry if I skip much details about sequence alignment, because tomorrow we are going to explain work from our group called GenASM, is where we accelerate a read alignment algorithm using PEM specialized accelerator. And this is the paper. Damla will present it. She's the first author of this work tomorrow, 16 October. Please make sure you attend this talk. So it will be during the lecture time. All right, so conclusion on going directions, read alignment can be substantially accelerated using computationally inexpensive and accurate pre-alignment filtering. And all the three directions are used by mappers today, but filtering has replaced alignment as the bottleneck because the pre-alignment filter will check everything that is going to the sequence alignment. And it will have the heavy workload and then minimal workload only for the sequence alignment. So pre-alignment filtering doesn't sacrifice any of the alignment capability and doesn't modify or replace the alignment step. So it will be great benefits if you can focus on pre-alignment filtering, accelerate it as much as possible, and then you either can flush the sequence alignment computation to the CPU side, or you can still accelerate it, as I mentioned, by having many filtering units that can accelerate sequence alignment. Now, what else can be done? So what if we got a new version of the reference genome? So this is another work from our group. Imagine that you already have a read set, you have a reference genome. Now the reference genome gets updated. We have a new version of the reference genome. Do we really need to repeat the mapping step? Repeat all mapping all reads to the new reference genome? And this is what we propose in this work, is that to translate the differences between the old reference and the new reference and only map the reads to certain region in the new reference. This again will accelerate the entire read mapping step significantly. To get more details about the paper, this is the GitHub, and you can find the paper on archive where we accelerate the overall pipeline by up to 16.4x. Now, where's read mapping going next? So again, are we achieving this goal? Can you analyze read mapping in less than 1 second? For example, you just put your finger, get a blood sample and sequence it, analyze it, and get to know who's the owner of this sequence, for example. Still, we are very far from there. Are we achieving intelligent genome analysis? Of course, no, because we're still very far from there. But again, the future is very bright for genome analysis. Now, we are still using conventional architecture to accelerate, even if we have specialized architecture, but we need to move the data from the sequencer itself to the storage, to the main memory, to the specialized architecture, or the CPU itself. And this is very expensive. And still we have open question where to process these genomic data. Now we know that most speed up comes from parallelism enabled by both of them, novel architecture and algorithms. Developing architecture alone is not enough. Developing algorithm alone is not enough. So we need to find both solution. And this is true for many applications from companies where they have specialized chip. And you already seen this in many of the lecture of Professor Onur Mutlu. So now the question remains, if these sequencing machine provide you genome long read, like one piece that is full sequence of your DNA, and 100% accurate, will that eliminate the need for read mapping? So I will ask you to rethink about metagenomics, where you have viruses, bacteria, where you still need to compare them to too many species. Even if you have the long sequence, you still don't know what's this. Is it bacteria, viruses, what type of bacteria? Even for pangenomics, or where you compare to humans, you still need to know what are the alterations that happens to the reference genome. Is it insertion, deletion, how large? So you still need to perform dynamic programming, hashing, pre-alignment filtering, and so on to know these efficiently. So read mapper or read mapping will never die or outdated, and we still use it even once the sequencing machine get 100% accurate and can generate genome long reads. Now, this is the last slide, sorry for being late. And system design for bioinformatics is a critical problem. It has large scientific, medical, societal, personal implications. And this lecture is about accelerating key steps in bioinformatics called genome sequence analysis or read mapping. In particular, we focus on read mapping throughout the major part of the lecture. Many bottlenecks still exist in accessing and manipulating a huge amount of genomic data during analysis. And after accelerating the read mapping and reduce its bottleneck, or we shift the bottleneck from read mapping to the next steps, then we need to shift our acceleration effort to these other steps as well. We cover various recent ideas from our lab, from other labs to accelerate read mapping. And this journey already started since September 2006 from Professor Onur Mutlu himself. I started 7 years ago, and we hope that we will continue accelerating genome analysis until we achieve an intelligent genome analysis. I would like to acknowledge many of my colleagues, funding agencies, and the source code of all the work that I presented, it's already public in this website. You can access it, you can download it and play with it. And these are two great resources for whoever is totally new to this topic, can get introduced from the fundamental, the basics, all the way to major research directions that we are working on. So if you are a student with ETH and you would like to work with us on any of these topics, please talk to me, send me an email. You can discuss with me anytime. And if you are outside ETH, then feel free to apply to our group to join working on bioinformatics and many other topics. So I would like to thank all of you. I'm so sorry to take 8 minutes more, but we have Piazza forum. Please send question there. I'm happy to take question now if you would like. I will stay for more minutes. And thank you so much for your patience. Have a very good day. Yeah, feel free to ask question. I will stay for more, for 5 minutes more.
Mohammed, I guess I'll make a couple of comments very quickly. Not a couple of comments, but thanks for the lecture. I assume people are hearing me, right?
Yes.
Yeah. So this is a great topic area. So if people are interested, feel free to talk with us, with me, Mohammed, and anyone else doing research in the group. Tomorrow we will cover GenASM, as Mohammed mentioned. This is one of the state-of-the-art works, actually. It's not even presented yet. It's officially going to be published next week at the micro conference. And tomorrow, actually, we're going to see several other works on accelerating machine learning, for example, accelerating sparse matrix computations and doing some in-DRAM data movement. So hopefully people will attend and see some other ongoing research for tomorrow. So today's lecture was best delivered, I think, by Mohammed, because Mohammed actually did his PhD with me and Can Alkan on exactly this topic, intelligent genome analysis, and he's continuing as a postdoc right now on this topic. So feel free to reach out to us if you have any more questions. This is in my mind an example of future application and algorithm architecture co-design that can really disrupt the systems, computing systems, as well as systems in general going into the future. And there are a lot of exciting directions. So if you're interested, this is the right time to jump onto the train, I would say. Okay, I think this is a good time to end, Mohammed. I don't see any questions either, unless you got some private ones. But let's hope that we get some good discussion on Piazza.
Thanks so much, Honor.